As the day settles and the body moves toward rest, cellular activity does not stop. It changes its rhythm. In this quiet shift, mitochondria adjust their work with a sense of timing that follows the body's daily cycle. This section will move gently through that idea. It will introduce mitochondria as adaptable energy systems that respond to night and day, especially to the restful, fasting-leaning conditions of the evening. The aim is to set a calm foundation, that energy production, repair, and signaling are paced by time. If attention drifts, that is welcome. The key ideas will return softly with unhurried repetition. Begin with the basic picture. Mitochondria create usable energy for the cell, mainly in the form of ATP through oxidative phosphorylation. They also handle redox balance, which refers to how electrons are moved and stored in molecules like NADH and NAD+. Over a 24-hour cycle, the demand for ATP rises and falls, and so does the preferred mix of fuels, such as carbohydrates and fats. At night, in many tissues, the body leans toward maintenance, conservation, and preparation. Mitochondria participate by modulating respiration, adjusting network shape, and renewing components. This is not a sharp switch, but a gradual realignment that matches a slower metabolic tempo. To understand why timing is involved, consider the circadian clock, the internal timing system that coordinates many processes over the day and night. The clock signals to cells when activity is expected and when recovery is preferred. Mitochondria listen to those signals through changes in gene expression, protein modification, and metabolite levels that also rise and fall across the cycle. At night, this coordination helps align substrate use with availability. When feeding pauses and the body is quietly resting, mitochondria often reduce high-intensity energy output and make room for inspection and repair. This rhythm helps ensure that resources are used efficiently and that stress on the system remains within safe bounds. Within this nightly alignment, respiration, the process of oxygen consumption to drive ATP production, can show time-of-day patterns. Some pathways become more prominent at certain times, while others recede, not as an on-off event, but as a smooth wave. The balance between carbohydrate oxidation and lipid oxidation can shift as hormones, nutrients, and the circadian clock set the stage. This gentle choreography reduces conflict between supply and demand. It also supports redox balance, ensuring that electrons move through the respiratory chain with minimal leakage, lowering unnecessary oxidative strain. Mitochondria are not static. They form a network that changes shape through fission and fusion. Fission separates segments which can help isolate parts that need removal, while fusion blends contents, sharing proteins and mitochondrial DNA across the network. These dynamic processes respond to the time of day. During evening and night, in many contexts, the network explores states that facilitate quality control and readiness for the next active period. This does not mean one state is always favored. Rather, a balanced rhythm across the 24-hour cycle maintains resilience. The details depend on tissue type, behavior, and nutritional patterns, but the theme is consistent. Timing matters. Another topic that gently fits into the evening picture is mitochondrial maintenance. Cells use mitophagy, a selective form of autophagy, to remove damaged mitochondrial parts, and they use biogenesis to replenish and rebuild. The interplay between fission, fusion, and mitophagy is like a maintenance sequence, done most efficiently when energy demands are lower. At night, conditions often favor inspection and turnover, with the circadian system providing cues that steer these processes. The following sections will return to these ideas in more depth, walking through how quality control and renewal are coordinated. Redox balance is also linked to timing. Molecules like NAD+ and ADH shift with feeding and fasting, and these shifts influence enzymes that edit mitochondrial proteins through processes like decetylation. This editing tunes respiration, stress defenses, and metabolic flexibility. At night, when the internal environment changes, these redox-linked signals help mitochondria adopt a calmer stance. The result is not silence, but an orderly slowing that makes space for stability. Quiet signals lead to deep effects, including protection from excessive oxidative byproducts. It is helpful to acknowledge that the exact nightly pattern is not identical in every organ or species. Day-active and night-active organisms organize their cycles differently. Muscles, liver, brain, and heart each have their own schedules and demands. Even so, the general principle holds: mitochondria are entrained by the larger rhythm of life, and in the evening many tissues prioritize tasks that sustain long-term performance. Thinking in this way is soothing for study. The details can be learned gradually, while the larger frame remains steady. As this series unfolds, the focus will shift gently from respiration to dynamics, then to redox, to quality control, and to the pathways that guide mitochondrial renewal. The terms fission, fusion, mitophagy, and biogenesis will become familiar. Their roles at night will become clear. There is no rush. Each idea will appear more than once, from slightly different angles, so that understanding can accumulate calmly. If the mind wanders, that is part of the design. Now that the foundation is laid, it is enough to hold a simple map in mind. Over the day, energy demand rises and falls. The circadian clock provides timing. Mitochondria align their respiration, their shape, their maintenance, and their signaling with that timing. At night, many of these processes lean toward conservation and repair. This quiet order supports the next day's work. It is a cycle, repeated and refined, every 24 hours. Before closing this section, a brief reassurance: precision will come with repetition. Hearing terms like oxidative phosphorylation, redox balance, and mitochondrial dynamics should feel comfortable with time. Each will be defined more fully later. For now, it is enough to know that mitochondria are not only power producers, they are time-aware caretakers of cellular energy and health. In the evening, they adjust with care. We will move next into a closer look at how the circadian system shapes mitochondrial behavior. From there, the script will explore how respiration follows daily tides, how morphology changes are used as tools, and how nighttime maintenance prepares tissues for the day ahead. The pace will remain steady and soft. The guidance will be consistent. If drowsiness appears, welcome it. The ideas will be here when attention returns, repeating like gentle waves, always familiar, always calm. As we move more deeply into the rhythm of evening, it helps to meet the quiet clock that keeps cellular time. The circadian clock is an internal timing system, present in nearly every cell, guiding when processes begin, when they pause, and when they prepare for the next cycle. Mitochondria, the cell's energy hubs, do not work at a steady, unchanging rate across the day. They listen to these timing cues and adjust. What follows is a calm orientation to the core clock components and how they shape mitochondrial behavior from light to dark and dark to light, aligning energy production, fuel choice, and maintenance with the predictable ebb and flow of daily life. If a phrase drifts by, that is fine. Ideas will return gently with the same unhurried cadence. At the center of circadian timekeeping are paired feedback loops of genes and proteins that rise and fall every 24 hours. Two proteins, often discussed together, form the activator core in many tissues, clock and BML1. They act in the nucleus, turning on sets of target genes, including two families called period PR1, PR2, PR3, and cryptochrome, Cry1, Cry2. As PR and Cry proteins accumulate, they return to the nucleus and reduce the activity of the clock PML1 pair, allowing the system to quiet and then reset. Alongside this core loop, additional regulators help stabilize the rhythm and tune its phase. The effect is a reliable cycle of gene expression and protein modification that repeats daily, with a phase appropriate to the tissue and the organism's activity pattern. Mitochondria are attentive to this rhythm. They respond to direct transcriptional programs, where clock-controlled genes influence proteins within the organelle, and they also respond to post-translational signals, where the chemical marks on proteins shift with time of day. This two-level guidance is important. Transcriptional control can set broad availability of components over hours, shaping which enzymes are present and in what amounts. Post-translational control can fine-tune activity within minutes, adjusting enzyme flux, membrane channel behavior, and the assembly state of respiratory complexes. In combination, these mechanisms allow mitochondria to match their output to the moment, a little more when movement and cognition rise, a little less when stillness and repair take priority. One calm way to picture this is through substrate preference. During the waking hours, when feeding is more likely, tissues may favor the use of carbohydrates in certain windows, then lean more toward lipid oxidation during others, depending on the tissue and the species' activity pattern. At night, during fasting or reduced intake, pathways of fatty acid oxidation may be more engaged in some organs, while others protect glucose for essential tasks. These are not abrupt changes. They are progressive shifts, shaped by hormones, nutrient levels, and the clock's influence on metabolic gene networks. The purpose is efficiency, reduce mismatch between supply and demand, lower unnecessary strain on the electron transport chain, and keep the redox environment within a comfortable range. Oscillations are not limited to fuel selection. Mitochondrial respiratory rates themselves can cycle with the day. Measurements in different models have shown time-of-day differences in oxygen consumption and in the capacity to oxidize particular substrates. These changes often mirror the proteomic landscape, where a meaningful fraction of mitochondrial proteins oscillates in abundance or modification across the 24-hour period. While the exact pattern depends on species, organ, and behavior, a consistent theme emerges. Mitochondria are not constant engines. They are rhythmic powerhouses, tuning their throughput and preference in step with a cellular schedule. The circadian system also coordinates the redox environment that mitochondria inhabit. Molecules such as NAD+ and NADH, which carry electrons and reflect cellular energy state, fluctuate with feeding and fasting and with clock control of the enzymes that make and recycle them. Because many mitochondrial and nuclear regulators sense NAD+ levels, these oscillations translate timing information into the regulation of desitylation and other modifications. As NAD+ rises at certain times, sirtuin enzymes become more active, adjusting the acetylation of mitochondrial proteins, transcription factors, and histones. This, in turn, guides oxidative phosphorylation, stress defenses, and metabolic flexibility across the day-night cycle. In evening conditions, these signals often help establish a quieter, more protective state, supporting maintenance while keeping essential ATP production steady. The clock's influence reaches the structure of the mitochondrial network as well. Mitochondria undergo fission and fusion, processes that change network shape and help maintain quality. There is evidence that elements of this machinery show time-of-day patterns. Proteins that promote fission and those that support fusion can vary in expression or modification across the cycle. This rhythmic tuning of dynamics helps align morphology with function. When energy demand is high, a more fused network can support efficient distribution of membrane potential and respiratory capacity. When maintenance tasks are prioritized, fission can help isolate segments for removal. Again, the rule is balance. Each phase has its role, and the clock gently allocates time to each. An important point for restful learning is that these rhythms exist across scales. At the whole body level, the central clock in the brain tracks light and helps set timing for behavior, hormones, temperature, and feeding patterns. In the periphery, each organ and even each cell has local clocks that respond to both central signals and local conditions. Mitochondria feel both influences. They align with systemic cues, like meal timing and sleep-wake cycles, and with cell-autonomous programs that determine when certain genes are transcribed or when a protein is modified. This layered organization creates reliable daily windows for peak performance, for careful restraint, and for repair. Because oxidative phosphorylation involves the controlled transfer of electrons, timing helps limit unnecessary production of reactive oxygen species. When substrate supply and respiratory capacity are synchronized, electrons move with fewer detours and antioxidant systems can keep pace. When activity and repair are placed into different parts of the day, they interfere less with each other. Over many cycles, this scheduling supports mitochondrial resilience, fewer cumulative insults, more regular opportunities to replace damaged components, and a steadier baseline from which to respond to stress. It is natural to wonder how strict these rhythms are. They are strong enough to be measured, but they are also adaptable. If feeding schedules change, the clock can shift. If light exposure differs, phase can adjust. Short-term changes may blur some peaks, but the system tends to re-establish order when cues stabilize. For learners, this means that the precise timing of a pathway's peak may vary by context, but the underlying principle remains sound. A daily pattern exists, and mitochondria are guided by it. Now that the architecture is in view, we can gently connect the pieces. Clock and BMAL. One initiate waves of gene expression that touch many mitochondrial functions. Person Sriye return as evening dampers, helping the system reset. Accessory regulators refine the phase and amplitude, keeping the rhythm stable. Downstream, mitochondrial proteins appear and recede on a schedule, and their activities are tuned by post-translational marks that also keep time. Substrate selection follows these cues, as do respiratory flux and redox signals. Dynamics, fission and fusion participate in the choreography, making maintenance efficient. And through it all, the goal is alignment. Place energy production where it is needed. Place repair where it fits best, and reduce friction between competing demands. A brief reinforcement may help the ideas settle. The circadian clock is a molecular metronome. It sets a pace for gene expression and protein state. Mitochondria respond by adjusting what they burn, how fast they work, and how they maintain themselves. This keeps energy efficient, preserves redox balance, and lowers avoidable oxidative stress. The pattern is not rigid, but it is reliable enough to prepare tissues for predictable cycles of feeding, activity, rest, and fasting. As we close this section, let the key points rest quietly. There is a clock in the cell. Its core components create a daily wave of instructions. Mitochondria read those instructions and adapt their function across the light-dark cycle. Certain fuels are preferred at certain times. Respiratory capacity and protein abundance can oscillate. Post-translational marks refine the timing further. The result is a calm efficiency that supports both performance and repair. In the next section, we will move more closely into respiratory rhythms themselves, how oxygen consumption and enzyme capacity change with time of day, and how this relates to the gentle switching of substrates. The pace will remain steady and soft. If attention drifts, the rhythm of the ideas will return, like a tide coming back to the same shore, familiar and unhurried. Settle into a calm understanding that respiration in the cell is not uniform across the day. It follows a gentle tide. Mitochondria shift how much oxygen they consume, which fuels they handle most easily, and how efficiently they pass electrons along the respiratory chain, all according to time of day. This section will move slowly through those patterns, focusing on the soft idea of capacity rising and falling like waves, rather than turning on and off. If a detail passes by, it will return later with the same quiet cadence. Across a 24-hour cycle, mitochondrial respiration can oscillate in a substrate-specific way. In some tissues, the capacity to oxidize carbohydrates reaches a crest during one part of the day, while the capacity to oxidize long-chain fatty acids peaks at another. These shifts do not happen abruptly. They emerge from upstream changes in proteins and enzymes, and from the signals that inform mitochondria about feeding, fasting, and rest. The result is a form of anticipatory tuning, where enzyme levels and activities are adjusted so that fuel preference and throughput are matched to predictable behavioral states. Part of this tuning is visible in the mitochondrial proteome. Many mitochondrial proteins rise and fall across the day, including rate-limiting enzymes that guide entry points into major pathways. When these proteins accumulate at specific times, the corresponding respiratory responses to their substrates also tend to be strongest in those windows. For instance, when proteins that gate fatty acid entry into mitochondria are more abundant, respiration supported by long-chain fatty acids tends to be higher. When components that process pyruvate are more available, carbohydrate-supported respiration is favored. This pattern, observed in carefully timed measurements, suggests that mitochondria keep time not only through gene expression, but through functional output that follows those molecular rhythms. These daily changes have also been seen directly in tissues. Human skeletal muscle, for example, shows a day-night rhythm in oxidative capacity, with higher respiration in the late evening compared to the early day in some protocols. This indicates that even without large changes in external activity, the tissue's intrinsic capacity can vary by clock time. Such measurements, taken under controlled conditions, reinforce the core idea: mitochondrial oxidative metabolism is not static. It is sculpted by time, reflecting a balance between expected demand and planned maintenance. The clock's influence extends beyond protein abundance to the chemical state of proteins. A key layer involves NAD+, a molecule that tracks cellular redox status and energy availability. NAD+ levels fluctuate with circadian control, and those fluctuations regulate deacetylases, including mitochondrial SIRT3. When NAD+ is higher, SIRT3 activity increases, leading to deacetylation of specific mitochondrial enzymes and antioxidant systems, which can enhance their function and adjust respiration to the appropriate level for that time of day. These cycles in NAD+ and enzyme acetylation contribute to rhythmic respiration and help align oxidative capacity with fasting and feeding windows, improving efficiency and reducing unnecessary oxidative strain. It may help to pause and reinforce the idea gently. Think of the cell preparing for the day's phases in anticipation of activity. Mitochondria organize a set of enzymes and cofactor states that support higher throughput for particular fuels. In anticipation of rest, they favor settings that conserve energy, maintain redox balance, and permit inspection and repair. The enzymes that process lipids or pyruvate do not simply appear. They follow a schedule guided by clock genes and by nutrient timing. In this schedule, respiration measured with different substrates shows distinct peaks, each corresponding to that substrate's favored window. The pattern is consistent across models: rhythms in proteins, rhythms in respiration, and a thread of clock control that ties them together. These rhythms matter because oxidative phosphorylation requires careful coordination. Electrons must flow smoothly through complex enzyme to IV, oxygen must be reduced efficiently, and ATP synthesis must keep pace without excessive buildup of intermediates. When respiration peaks align with substrate availability and metabolic need, the electron transport chain operates with fewer detours, and reactive oxygen species are more easily managed by antioxidant systems. With regular windows for higher flux and separate windows for maintenance, mitochondria experience fewer conflicts between producing energy and repairing machinery. Over many cycles, this separation supports stability. Another gentle layer is the observation that circadian disruption can flatten or shift these oscillations. When core clock components are impaired, mitochondrial respiration and enzyme rhythms become blunted, and substrate-specific peaks may be lost or diminished. This does not mean respiration stops, rather, its fine-tuned timing is less distinct, which can reduce metabolic efficiency. Conversely, reinforcing regular cycles of light, feeding, and rest can help re-establish clear peaks and troughs. In this way, the clock communicates timing to mitochondria through transcriptional programs, post-translational edits, and redox cues, and mitochondria respond by shaping their daily respiratory profile. NAD plus oscillations sit at the center of this communication. The clock influences the enzymes that make and recycle NAD+, creating daily waves of availability. In response, deacetylases, like SIRT3, modulate the acetylation state of oxidative enzymes, antioxidant proteins, and components of intermediary metabolism. The outcome is a time-stamped configuration of mitochondrial function. At some times, deacetylation promotes fatty acid oxidation and supports fasting metabolism. At others, different configurations may be preferred. This mechanism allows relatively rapid adjustment without the need to rebuild the proteome each hour, adding flexibility to the deeper transcriptional cycles. It is natural to ask whether these oscillations are large or small. They are measurable and meaningful, but designed to be smooth. The body prefers stability. Peaks and troughs are shaped to avoid abrupt transitions, keeping ATP supply adequate and membrane potential steady. The gentle wave image is appropriate, a rise that prepares the cell for activity, a fall that makes room for restoration, and a constant attention to redox safety. Across tissues, the exact timing and amplitude differ, reflecting local demands in the organism's behavior, but the organizing principle endures. Before closing, a soft recap may help the ideas settle. Respiratory capacity changes with time of day. Mitochondrial proteins, including rate-limiting enzymes for major fuels, oscillate in abundance, and respiration measured with those fuels shows corresponding daily peaks. These patterns match behavioral states like activity and rest. The clock guides this process through gene expression and through redox-linked deacetylation, especially via NAD+, sensitive enzymes such as SIRT3. The result is efficient oxidative metabolism, lower unnecessary oxidative stress, and protected windows for maintenance. As the section ends, let the image of a dim, orderly laboratory rest in mind. Instruments are not turned off, they are tuned, dials are turned slightly up for one pathway, slightly down for another. Through the evening and night, capacity is conserved, electrons are shepherded carefully, and the network prepares gently for the next day's work. In the following section, we will move calmly into the choreography of mitochondrial dynamics, how fission and fusion change with time, and how morphology supports these daily flows in respiration and repair. Settle into a calm understanding that mitochondria are not fixed shapes inside the cell. They move through a quiet choreography of separation and joining that supports energy balance and repair across the day and night cycle. This choreography is known as mitochondrial dynamics, fission, the process of dividing one mitochondrion into smaller units, and fusion, the process of merging mitochondria into a more continuous network. The rhythm of these processes is guided in part by the circadian clock, creating daily patterns in morphology that help align energy production, membrane potential, and quality control with expected phases of activity and rest. Fission is largely driven by a protein called DRP1, which is recruited to the outer mitochondrial membrane and constricts it, forming separate mitochondrial fragments. Fusion depends on mitofusin's MFN1 and MFN2 on the outer membrane, and OPA1 on the inner membrane, which together enable the mixing of contents and maintenance of cristae structure. Across the 24-hour cycle, the activity states of these proteins can change, shifting the balance between fragmentation and elongation of the network in a time-of-day dependent manner. These shifts are not abrupt. They are gentle and adaptive, tuned by the clock to keep mitochondria prepared for alternating periods of demand and recovery. A central finding is that DRP1's activity follows circadian cues through changes in its phosphorylation state, which rise and fall over the day and night. When DRP1 is phosphorylated at specific sites, its recruitment and constriction activity are modulated, influencing when fission is favored and when it is restrained. Upstream regulators including calcineurin, a phosphatase whose activation can be circadian, adjust DRP1's phosphorylation, thereby shaping daily patterns of mitochondrial morphology and the cell's readiness for quality control. When DRP1 rhythms are disrupted, the normal oscillation of mitochondrial shape and bioenergetics is blunted and ATP production loses its diurnal pattern. Fusion proteins also participate in this temporal alignment. MEFN1/2 and OPA1 show regulation by clock-linked pathways, including sirtuin-dependent dacetylation, which can adjust OPA1's function and support healthy inner membrane fusion during appropriate windows. This means the network can become more tubular when coordinated distribution of membrane potential and respiratory capacity is helpful, and more fragmented when isolating portions for mitophagy becomes a priority. In this way, dynamics serve both performance and maintenance, guided by a schedule that prioritizes efficiency by day and careful housekeeping at night. These rhythmic changes have clear functional consequences. More fused networks tend to support stable membrane potential and efficient electron transport, which can be advantageous when sustained ATP delivery is required. Increased fission can make it easier for the cell to identify and segregate damaged regions, marking them for removal through mitophagy when conditions favor repair. The readiness for mitophagy itself appears to be clock-sensitive. Regulators such as PINK1, Parkin, and BNIP3 can exhibit daily patterns of expression or control that align with the timing of mitochondrial turnover. When clock genes like BMAL1 or clock are disrupted, these rhythms flatten, mitophagy markers can diminish, and mitochondrial fragmentation may rise in a maladaptive way, with reductions in membrane potential and increased oxidative stress. The mechanisms that connect the circadian clock to dynamics are layered. At the transcriptional level, clock-controlled factors influence the abundance of fission and fusion machinery over the day. At the post-translational level, enzymes responsive to cellular energy state and redox, such as AMPK and sirtuins, modulate phosphorylation and acetylation of dynamics proteins and their regulators, shifting morphology in sync with feeding, fasting, and rest activity cycles. For example, AMPK can promote fission during energy stress by recruiting DRP1 to mitochondria, but can also favor fusion through increased MFN2 expression under fasting conditions, enhancing ATP efficiency when it is most needed. Sirtuin activity, linked to oscillations in NAD+, can decetylate OPA1 and support balanced fusion that protects network integrity. These daily patterns are seen in diverse models. Cultured hepatocytes and fibroblasts show rhythmic transitions from more tubular networks to more fragmented states over circadian time, in step with changes in respiration and ATP levels. In tissues, loss of clock components diminishes the normal oscillation of mitochondrial morphology, leading to more static, often fragmented networks and functional compromise. Together, these findings support a consistent view. The clock does not simply tell mitochondria when to work, it helps determine how they are shaped to do that work, and when they are shaped to rebuild. Importantly, balance is the theme. Excessive fission can lead to persistent fragmentation, decreased membrane potential, and vulnerability to stress, while excessive fusion can mask localized damage and delay clearance of impaired regions. When rhythms are intact, the system avoids both extremes, allowing the network to elongate when continuity is beneficial and to segment when renewal is scheduled. Over many cycles, this balance supports resilience, fewer conflicts between energy production and repair, steadier redox homeostasis, and a healthier pool of mitochondria prepared for the next day's demands. As the organism transitions from wake to sleep, this choreography becomes particularly meaningful. A gentle increase in fission-associated readiness can support surveillance and mitophagy, while maintained fusion capacity preserves essential ATP production for baseline needs. Membrane potential tends to be more stable when morphology and respiration are aligned with clock time, and antioxidant systems can keep pace more effectively when maintenance does not compete with peak output. If the rhythm is perturbed by irregular light feeding or genetic disruption, morphology rhythms can flatten, mitophagy can be compromised, and oxidative load may rise, underscoring the value of temporal order. A brief, quiet reinforcement can help the ideas settle. Fission and fusion are not opposites in conflict. They are partners in care. DRP1-mediated fission tuned by circadian phosphorylation allows selective pruning. MFN1/2 and OPA1-mediated fusion maintains continuity and supports performance. Clock inputs arrive through gene expression and through energy and redox-sensitive enzymes, shaping when each process takes the lead. The result is a daily pattern of morphology that protects function, a network that breathes in and out, elongating to distribute energy, fragmenting to renew, always aiming for balance. Let this section close softly, like tracing a constellation's outline without rushing. The shapes inside cells change with time, and in that changing shape lies a quiet strategy for health. In the next section, we will rest gently into the signals that accompany these shapes at night, the membrane potential and redox patterns that rise and fall with the same patient rhythm, guiding mitochondria towards stability and calm efficiency. As evening settles across the internal landscape, mitochondria adjust their electrical and chemical tone with a timing that follows the day-night cycle, creating gentle patterns in membrane potential and redox state that support stability and calm efficiency through the night. These patterns are coordinated by the circadian system, which guides how energy is produced, how electrons are handled, and when protective processes are emphasized, allowing cells to conserve resources and prepare for the next active phase. It can help to imagine these signals as small, steady lights, voltage gradients and redox ratios, quietly maintaining order while the organism rests. Mitochondrial membrane potential. The voltage across the inner mitochondrial membrane shows time of day rhythms in central pacemaker regions such as the suprachiasmatic nucleus, where mitochondrial activity is higher during the light phase in many models, reflecting coordinated metabolic readiness. In these neurons, daily oscillations in excitability and ion handling accompany metabolic oscillations, forming a linked rhythm between electrical activity and mitochondrial function. When light cues and clock signals align, mitochondrial morphology and bioenergetics in the pacemaker region show clear daily structure, supporting precise timing for downstream tissues. Across systems, membrane potential tends to track with mitochondrial network state. More fused networks often maintain a stable, robust potential suited for sustained ATP support, while transient increases in fission facilitate surveillance and targeted turnover, especially as the organism transitions towards sleep. Time of day differences in fusion and fission proteins, including MFN2 and OPA1 for fusion and regulators of fission, shape these morphology, potential relationships in a circadian manner. When dynamics are perturbed, for example, with loss of MFN2 in SCN neurons, the usual diurnal patterning of mitochondrial shape and network function is diminished, underscoring how structural rhythms help stabilize bioenergetics across the cycle. Redox cofactors provide a second, interwoven layer of timing, as NAD+ and related nucleotide pools oscillate with the clock and feeding, fasting cycles, translating time into enzymatic activity that fine-tunes respiration and antioxidant capacity. A clock-controlled rise in NAD+ activates sirtuin deacetylases, including mitochondrial SIRT3, which modulate respiratory enzymes and antioxidant defenses to align oxidative capacity with the current phase, often favoring protective configurations at night. This biochemical timing offers flexibility while proteomes change over hours. Redox-sensitive modifications allow faster, gentle adjustments to keep the electron transport chain balanced and membrane potential steady. In central clock tissue and peripheral organs, rhythmic redox states are measurable, with SCN slices showing oscillations in cellular redox and metabolic readouts that mirror the behavioral cycle. In the liver and other metabolically active tissues, the clock drives rhythmic expression of enzymes in the NAD plus salvage pathway, ensuring that cofactor availability rises and falls predictably to support time-appropriate mitochondrial function. When this NAD plus linked rhythm is disrupted through genetic clock perturbations, mitochondrial oxidative capacity falls, but can be partially restored by replenishing NAD plus precursors, highlighting the causal bridge between redox timing and respiratory performance. Reactive oxygen species, or ROS, sit within this nighttime conversation not as purely harmful byproducts, but as controlled signals that help adjust metabolism and reinforce clock alignment when kept within physiological bounds. The circadian system coordinates both ROS production and detoxification by timing nutrient flux, uncoupling and antioxidant defenses, thereby limiting oxidative stress while preserving ROS's role as a messenger. When feeding or activity occurs at an unexpected time, ROS can rise when defenses are not optimally primed, reinforcing the importance of temporal order for redox homeostasis. Through the night, antioxidant pathways, including those governed by NRF2 and glutathione metabolism, are clock-linked, providing windows where detoxification is especially effective and synchronized to the expected metabolic load. Peroxide-doxin oxidation cycles, which reflect tissue redox state, also follow 24-hour rhythms, illustrating an ancient conserved layer of redox timing that complements mitochondrial control of electrons and potential. These synchronized systems allow mitochondria to maintain a high-quality signal environment in which membrane potential remains stable and oxidative stress is buffered, without suppressing useful ROS signaling. In pacemaker neurons, the coupling between high daytime excitability and mitochondrial readiness highlights how electrical and metabolic rhythms co-stabilize. Membrane potential and ATP demand are matched, and redox systems are poised to manage byproducts efficiently. During the dark phase, when activity wanes for diurnal species, a modest shift towards surveillance, supported by dynamics that permit selective fission, can occur without compromising essential potential, ensuring that mitophagy can proceed as needed. In constant darkness or with clock gene disruption, these coordinated patterns flatten, leading to altered mitochondrial shapes and potentially less efficient membrane potential control, a reminder of how environmental cues and internal clocks collaborate to preserve rhythm. A gentle reinforcement may help these ideas settle. Membrane potential is the quiet voltage of mitochondrial life, sustained by electron flow that is most efficient when timing aligns with substrate supply and cellular need. Redox cofactors, especially NAD+, encode time into enzyme states, regulating decetylation of respiratory and antioxidant proteins to keep potential steady and ROS signaling informative rather than damaging. Dynamics connect structure to function. Fusion stabilizes potential and distribution of capacity, while timely fission supports inspection and renewal, both under circadian influence. When the system is aligned, light cues consistent, feeding within expected windows, sleep allowing maintenance, membrane potential drifts calmly within its optimal range, and redox defenses are ready to absorb variation without strain. If the pattern is disrupted, potential can become less stable, ROS may increase at the wrong time, and antioxidant readiness may lag, though re-establishing regular cues often restores the rhythm. Over many cycles, this nightly coordination preserves mitochondrial resilience, reduces cumulative oxidative burden, and sustains a network prepared for tomorrow's work. Let the image be simple and quiet. Tiny voltage gradients glowing like candlelight and redox ratios breathing in time with the clock, together guiding mitochondria through the night with steadiness and care. In the next section, the focus will move gently to nighttime maintenance, how quality control and mitophagy use this calm environment to renew the network before dawn. As the body settles into evening rhythms, cells enter a quiet phase of care where maintenance becomes a gentle priority and energy production continues at a calm, sufficient pace. Within this window, mitochondria are surveyed for signs of wear, damaged segments are set aside, and renewal pathways are coordinated to keep the network healthy for the next day. This section invites an unhurried walk through that process. The pacing is slow, the language is soft, and the goal is clarity without urgency. Fission and fusion working together, mitophagy selecting what must be cleared, and biogenesis preparing what will replace it. Even under stress, cells prefer order over haste, and the circadian system helps schedule these tasks so they do not compete with peak activity. Begin with the choreography. Mitochondria change shape through two complementary processes, fission and fusion. Fission, driven predominantly by DRP1, divides the network, creating smaller units that can be examined more closely. Fusion, coordinated by MFN1 and MFN2 on the outer membrane and OPA1 on the inner membrane, merges mitochondria and redistributes proteins, lipids, and mitochondrial DNA, preserving function and buffering local defects. Across the day, these dynamics follow time cues. During active periods, a more connected network can support steady membrane potential and efficient ATP production. As night approaches, a modest increase in fission readiness allows the cell to isolate segments that are not performing well, guiding them towards selective removal. Fusion does not disappear. It remains available to maintain continuity and to prevent unnecessary loss. Balance is the constant aim. Selective removal of damaged mitochondria proceeds through mitophagy, a specialized form of autophagy. One well-studied pathway begins when the mitochondrial membrane potential falls persistently on a subpopulation of organelles. PINK1, a kinase that normally transits and is degraded, accumulates on the outer membrane of these impaired mitochondria. There, it phosphorylates both ubiquitin and the E3 ligase parkin, activating parkin and recruiting it to the damaged organelle. Parkin deposits ubiquitin chains on outer membrane proteins, creating a signal for autophagy receptors and adapters. These in turn engage the autophagosome machinery, allowing the damaged mitochondrion or fragment to be enveloped and delivered to lysosomes for degradation and recycling. This PINK1, Parkin sequence is not the only route. Under hypoxia, or in tissues with specific cues, receptor-mediated mitophagy can proceed through proteins embedded in the outer mitochondrial membrane, such as BNIP3, NICs also called BNIP3L, and FUNCS1. These receptors carry LC3 interacting regions that link mitochondria to autophagosomes directly, often responding to shifts in phosphorylation states that mark stress or energetic need. They also interface with dynamics. BNIP3 can influence DRP1 activity and OPA1 function, favoring controlled fission when removal is required. FUNCS1 can modulate fission, fusion balance and participates in organizing the sites where mitochondria and the endoplasmic reticulum meet to coordinate turnover. Fission serves as an upstream organizer for mitophagy by segregating subregions with lower membrane potential or impaired respiration. Fission makes it easier to target removal precisely, conserving healthy network elements. Downstream autophagy adapters recognize ubiquitin tags or receptor motifs and build the autophagosomal membrane around the marked fragment. This sequence is more efficient when energy demand is modest and antioxidant defenses are steady, conditions that often coincide with the night for diurnal organisms. The circadian system contributes at several levels. It influences the abundance and modification of fission and fusion proteins. It shapes the expression and activity of autophagy genes, and it arranges redox and NAD+ linked signals that fine-tune when enzymes and receptors are most capable of acting with minimal collateral stress. When these rhythms are intact, the cell can survey, tag, and recycle without compromising baseline ATP needs. The endoplasmic reticulum, participates closely in this maintenance. Points of close contact between and mitochondria, often called mitochondria-associated membranes, or MAMs, serve as hubs for lipid exchange, calcium signaling, and the assembly of fission machinery. Under strain, including sleep loss or extended wakefulness, cells can increase the number of these contact sites, likely as a compensatory response to support trafficking, stress responses, and turnover. This structural adjustment consolidates during recovery sleep, when metabolic pressure eases and maintenance can proceed more fully. The ER helps position the fission apparatus, supplies lipids for autophagosome formation, and coordinates calcium signals that influence both mitochondrial dynamics and autophagy readiness. Mitophagy does not act in isolation. It is paired with biogenesis, the creation of new mitochondria and the replenishment of the network. After damaged components are cleared, transcriptional programs governed by co-activators such as PGC, 1-alpha, with partners like NRF1, NRF2, and TFAM, help rebuild respiratory capacity and support mtDNA maintenance. This sequence resembles careful pruning followed by quiet growth. At night, signals from energy sensors and sirtuins linked to predictable shifts in NAD+ and AMP levels can favor the activation of these renewal pathways at a time when they will not compete with peak energetic demand. Over repeated cycles, this balance between clearance and replacement maintains a healthy, adaptable mitochondrial population prepared for the next day's tasks. The preference for order shows most clearly when rhythms are disturbed. If clock timing is disrupted, the coordination between dynamics, mitophagy, and biogenesis can blur. Excess fission without appropriate clearance may yield persistent fragmentation and lower membrane potential, while insufficient fission can allow damaged segments to remain embedded in the network, increasing oxidative burden. Similarly, if turnover is attempted during peak demand, competing priorities can reduce efficiency. Reestablishing consistent cues, light-dark cycles, feeding windows, and sleep often restores more distinct windows for maintenance and performance, allowing mitochondria to return to their balanced schedule. It is helpful to revisit the roles of the main players, softly and without rush. DRP1 guided fission prepares segments for inspection. MFN1/2 and OPA1 support fusion, preserving membrane potential and sharing components to dilute minor defects. PINK1 senses a drop in potential and stabilizes on the outer membrane of compromised mitochondria. Parkin, activated by PINK1-mediated phosphorylation, builds ubiquitin signals that recruit autophagy receptors and membrane assembly. BNIP3, NSIX and FUNTS, one offer parallel or complementary routes responding to hypoxia, developmental transitions, or stress states, often with their own phosphorylation codes. The ER increases contact with mitochondria under load, guiding where fission occurs and supplying lipid building blocks for autophagosome membranes. Downstream, lysosomes complete the process, and transcriptional programs then restore capacity. Throughout this, the tone of the system remains calm. Mitophagy is selective. It does not remove more than is needed. Fusion is preserved. It does not overwhelm surveillance. Antioxidant systems remain engaged. They are not suppressed. The aim is continuity. Maintain enough ATP, keep membrane potential steady, clear what is beyond repair, and quietly replace what was lost. Nighttime provides a natural setting for this continuity, and sleep allows consolidation, especially after strain. If attention wanders, it is enough to remember that cells prefer planned care over last-minute response, and that mitochondria, guided by time, practice this care every night. As the section closes, let the sequence rest in a simple outline. Survey gently, separate what needs attention, mark what must be recycled, clear with precision, rebuild with care. The network emerges steadier and ready for tomorrow. In the next part, we will ease into how biogenesis is cued toward dawn, and how renewal completes the cycle, linking the quiet work of the night to the energetic needs of the day. As the night's maintenance winds down, cells begin a quiet phase of rebuilding that prepares the mitochondrial network for the day ahead. This renewal is called mitochondrial biogenesis, and it unfolds with calm precision under the guidance of a COL activator named PGC-1-alpha. In gentle coordination with transcription factors and energy sensors, PGC-1-alpha helps increase mitochondrial content, restore respiratory capacity, and support mtDNA care, so that the system that powers cellular life starts the next cycle steady and prepared. Think of this as measured growth after careful pruning, a replenishment that follows inspection and selective clearance, arriving on time with the circadian schedule and the ebb of metabolic pressure from the night's tasks. PGC-1-alpha, short for peroxisome proliferator, activated receptor gamma coactivator. Its most familiar partners in biogenesis are NRF1 and NRF2, transcription factors that amplify the expression of nuclear genes encoding respiratory chain subunits, import machinery, and essential factors for mitochondrial DNA care. Downstream, NRF1/2 increase transcription of TFAM, the mitochondrial transcription factor that binds mtDNA, supports its packaging, and is crucial for mtDNA replication and transcription, anchoring genome maintenance as new organelles are formed. Through this axis, PGC-1-alpha to NRF1/NRF2 to TFAM, cells coordinate the production and import of mitochondrial proteins with the replication and expression of the mitochondrial genome, distributing fresh capacity across the network in a way that is both comprehensive and restrained. This rebuilding is not random in time. Energy sensing pathways align biogenesis with feeding, fasting cycles, and circadian cues to ensure renewal occurs when it will not compete with peak output. AMPK, a kinase that senses cellular AMP cell ATP balance, and SIRT1, a NAD+ dependent deacetylase, converge on PGC-1-alpha to activate it, one by phosphorylation and the other by deacetylation. When fasting and the circadian clock elevate NAD+ via the NMP salvage pathway, SIRT1 activity rises and enhances PGC-1-alpha function. When energy is scarce and AMPK is engaged, PGC-1-alpha is likewise stimulated, linking energy state to gene programs that support new mitochondria. These same loops are intertwined with the clock itself, as oscillations in NMP and NAD+ propagate timing information to SIRT1 and onward to PGC-1-alpha, integrating metabolic signals with circadian structure so that biogenesis can be scheduled with reliable phase relationships. The result is a life cycle that pairs clearance with replacement, after periods of heightened mitophagy, often supported by fission and selective tagging of damaged fragments. PGC-1-alpha driven transcription restores capacity and balances the pool of mitochondria, keeping overall function steady across days. Crosstalk helps coordinate these opposing flows. Parkin, a mitophagy E3 ligase, can relieve repression of PGC-1-alpha by targeting the PGC-1-alpha suppressor Paris for degradation, linking the need for removal to the permission to rebuild. Other connections exist as well, such as PGC-1-alpha's influence on mitophagy-related genes through NRF1 and related partners, underscoring that quality control and biogenesis are not separate silos, but communicating arms of a unified maintenance plan. Within this plan, TFAM has a special place, as NRF1/2 raise TFAM expression. mtDNA gains the support required for accurate replication and transcription, ensuring that new or refreshed mitochondria carry intact genetic instructions. Because many respiratory subunits are encoded in the nucleus, while critical core subunits are encoded in mtDNA, biogenesis must synchronize two genomes. The PGC-1-alpha, NRF, TFAM axis supplies that synchronization, guiding coordinated expression, import, and genome packaging to create functional respiratory complexes at the right time. This reduces mismatches between protein availability and assembly demand, lowering stress on the electron transport chain as the day's requirements rise. Circadian alignment adds an additional layer of calm control. In peripheral tissues like liver and muscle, clock components shape the timing of mitochondrial gene programs, aiding the daily coupling of protein abundance, morphology, and turnover with expected behavior. Through the NAMP, NEDI+, SIRT1 axis, clock timing modulates PGC-1-alpha activity, aligning biogenesis with late night or early day windows, depending on tissue and species, when the network can incorporate new capacity without disrupting essential homeostasis. When rhythms are disturbed, these patterns may flatten, biogenesis can become mistimed relative to mitophagy, and respiratory efficiency may fall. Restored regularity, stable light cues, consistent feeding windows, adequate sleep tends to bring back clearer windows for renewal and the quiet return of balance. The gentleness of biogenesis reflects its scope, rather than large bursts. Cells often favor modest, repeated increments that preserve steady function while gradually refreshing the network. PGC-1-alpha can also cooperate with antioxidant pathways, including NERV2-linked programs, supporting redox resilience as new capacity is installed, an interplay that has been observed across contexts and tissues. In this way, replenishment is paired with protection as respiratory potential increases. Antioxidant defenses and genome maintenance keep pace, containing oxidative byproducts and preserving the stability of the emerging pool. It may be reassuring to revisit how energy sensing guides this sequence. AMPK activation during energetic strain signals that efficiency and renewal are needed. SIRT1 activation during NAD plus peaks signals that timing and transcriptional remodeling can proceed. Together, they lift PGC-1-alpha into an active state, opening access to NRF1/2 and TFM, and to a wide set of nuclear-encoded mitochondrial genes that support import assembly and oxidative phosphorylation. Because NAD+ and AMPK activity are themselves rhythmic with feeding, fasting, and the clock, the system naturally places biogenesis into predictable windows. This predictability lowers competition with peak ATP demand and reduces the risk that assembly and repair will clash with high respiratory flux. As with earlier sections, a soft recap can help the ideas settle. Mitochondrial biogenesis is a calm rebuilding that follows selective clearance. PGC-1-alpha is the central COSA activator, partnering with NRF1 and NRF2 to raise TFM and a broad suite of mitochondrial genes, coordinating nuclear and mitochondrial genomes to produce functional respiratory capacity. Energy sensors, AMPK and SIRT1 activate PGC-1-alpha in response to cellular state and circadian NAD plus rhythms, placing renewal into suitable night or dawnward windows, where the network can integrate new components without strain. Cross-talk with mitophagy ensures that what is removed is gradually replaced, preserving total function and lowering cumulative oxidative burden over many cycles. If attention has drifted, it is enough to hold a simple picture. During the night, inspection and selective removal create space. As dawn approaches, PGC-1-alpha opens the transcriptional gates for new capacity. NRF1/NRF2 rise. TFM supports mtDNA replication and transcription, and nuclear-encoded proteins flow toward assembly. AMPK and SIRT1 provide the timing signal, shaped by fasting, feeding, and circadian NAD+. The outcome is balance. Like careful gardeners who prune and then nurture growth, cells clear and then rebuild with measured timing, so the mitochondrial fleet is ready for the day. We will continue gently from here. The next sections will touch the timing signals that converse with PGC-1-alpha, NAD plus rhythms and sirtuin activity, and how these signals fine-tune mitochondrial proteins across the cycle. The pace will remain quiet and steady. Concepts will return with soft repetition, allowing understanding to grow without effort, as the network renews itself in the background, preparing for another calm beginning. Move gently into the idea that timekeeping and metabolism are not separate threads, but a woven fabric within the cell, where signals of day and night shape the availability of key molecules and the behavior of enzymes. At the center of this conversation is NAD+, a cofactor that rises and falls across the 24-hour cycle, and the sirtuin family of deacetylases, which depend on NAD+ to adjust protein function quietly and precisely. When NAD+ oscillates, sirtuin activity follows, linking feeding, fasting states and circadian cues to the tuning of mitochondrial proteins, chromatin, and transcriptional programs that guide oxidative capacity and stress resilience. The circadian clock helps set this rhythm by regulating the NAD+ salvage pathway, especially the enzyme NAMP, which is rate-limiting for NAD+ synthesis in many tissues. Clock and BMAL1, the core transcriptional drivers of the clock, promote daily waves of NAMPT expression, leading to predictable rises in NAD+ that activate SIRT1 and other sirtuins at the right times of day. In turn, SIRT1 feeds back to the clockwork by deacetylating BMAL1 and PER proteins and modulating clock. BMAL1-dependent transcription, closing a loop in which timing and metabolism reinforce each other with steady cadence. This interlocked feedback allows cells to translate energy state into gene expression patterns without abrupt transitions, maintaining continuity across cycles. Sirtuins connect this timing directly to mitochondria. SIRT3, the principal mitochondrial deacetylase, modulates the acetylation state and thus the activity of many enzymes in the respiratory chain, fatty acid oxidation, amino acid metabolism, and antioxidant defense. When NAD+ availability is higher, SIRT3 deacetylation generally supports more efficient oxidative reactions and enhances defenses that keep reactive oxygen species within physiological bounds, protecting membrane potential and preserving ATP output with minimal strain. Because NAD+ itself cycles under clock control, mitochondrial function receives daily, redox-linked adjustments that align substrate preference and flux with expected phases of feeding and fasting. The outcome is smoother electron flow, more stable membrane potential, and fewer conflicts between production and repair. SIRT1 acts in a complementary space, bridging nuclear programs and mitochondrial needs. When NAD+ rises, SIRT1 deacetylates and activates PGC-1-alpha, a co-activator that drives transcription of mitochondrial genes through NRF1/NRF2 and TFM, supporting biogenesis and capacity renewal, at times when it will not compete with peak energy demand. This places renewal into windows shaped by both circadian and nutrient cues, often aligning with late night or dawn periods, depending on tissue and behavior. In addition, SIRT1's interaction with clock components in the suprachiasmatic nucleus and peripheral tissues helps maintain robust rhythms in gene expression, ensuring that NAD+ cycles and sirtuin activity remain phase-appropriate. In this way, the clock writes the schedule, NAD+ carries the timing ink, and sirtuins annotate proteins and chromatin to match the hour. Feeding and fasting patterns are part of the same quiet dialogue. Nutrient state influences the redox environment, shaping NAD+ levels and thereby sirtuin activity. Fasting and low-energy signals tend to favor higher NAD+ and greater SIRT1/SIRT3 engagement, which encourages fatty acid oxidation, supports antioxidant systems, and primes biogenesis through PGC-1-alpha when appropriate. The result is time-of-day differences in oxidative capacity and substrate use, with sirtuins helping to open and close these functional harbors on schedule. When these cycles are synchronized with behavior, regular light exposure, consistent meal timing, and restorative sleep, the mitochondrial network experiences fewer mismatches, and oxidative stress remains within manageable ranges. This conversation is also adaptive. If rhythms are perturbed, NAD+ cycles can flatten and sirtuin activity may drift out of phase, reducing the clarity of mitochondrial oscillations and the efficiency of downstream pathways. Yet, restoring rhythmic cues can rebuild amplitude, reinforcing the NAD+ loop, re-establishes circadian timing, improves the coherence of clock gene expression, and steadies mitochondrial function across the day-night cycle. Studies underscore that this is tissue-specific and context-dependent, but the central principle remains durable. NAD+-dependent circadian activity is a tunable interface where timekeeping shapes metabolism, and metabolism feeds back to sustained timekeeping. A brief calm reinforcement may help the ideas settle. NAD+ is not simply a static cofactor. It rises and falls with the clock and with feeding, fasting, guiding search-ins to deacetylate proteins when it best serves the cell. SIRT1 connects these NAD+ surges to clock machinery and to transcriptional programs that prepare mitochondria for the coming phase. SIRT3 carries the signal into the organelle itself, adjusting enzymes that govern fuel oxidation, respiratory efficiency, and antioxidant readiness. Together, they keep oxidative capacity, substrate preference, and stress defenses aligned with predictable rhythms, minimizing friction between the needs of the night and the demands of the day. Let the imagery be soft. Picture dim lanterns lining a quiet corridor, NAD+ levels rising like gentle light, sirtuins responding with small, precise edits that guide proteins, pathways, and organelles to their proper places in time. Nothing abrupt, nothing loud, only steady coordination between the cellular clock and its metabolic partners. In the next section, we will ease into how these timekeeping signals translate into practical shifts in fuel handling, how mitochondria favor carbohydrates or lipids at different times, and how this supports the broader stability of the network through each 24-hour cycle. Settle into the idea that mitochondria do not treat all fuels the same at every hour. They adjust preference with time, creating gentle peaks for carbohydrate handling and for lipid oxidation that align with daily rhythms of feeding, fasting, rest, and activity. These changes are not abrupt switches, but slow movements of capacity guided by the circadian clock and by nutrient timing, so that supply meets demand with minimal strain on the respiratory chain. Across tissues, proteomic rhythms and post-translational edits quietly reshape pathways, allowing mitochondria to favor one substrate window and then another, like dials that turn gradually through the day and night. Evidence for this time-structured selection is clear in studies that measured mitochondrial respiration with different fuels across 24 hours, showing substrate-specific peaks that depend on intact clock function. In mouse liver and other tissues, rate-limiting enzymes for fatty acid entry and for pyruvate processing oscillate, and these protein waves align with higher respiration on the matching substrate at predictable times of day. When the clock proteins PER1 and PER2 are removed, many of these rhythms flatten, and the diurnal pattern of nutrient-specific respiration is blunted, indicating a direct role for PER proteins in organizing fuel choice across the cycle. These findings suggest that mitochondria anticipate behavioral states, activity, rest, feeding, fasting, and prepare enzymatic capacity accordingly. Lipid handling provides a gentle example of phase-specific tuning. In clock-aligned conditions, components of mitochondrial fatty acid oxidation show daily oscillations in abundance and function, with higher capacity concentrated in discrete light, dark windows that coordinate with feeding, fasting patterns in the organism. The carnitine shuttle and downstream beta-oxidation enzymes can rise in concert, producing a time when long-chain fatty acids are processed more efficiently and with less oxidative strain. If feeding is shifted or composition changes, the phase of these lipid rhythms can shift as well, reflecting the interplay between external cues and the internal clock. In contrast, disruption of PR1/2 or short-term high-fat diet can blunt these oscillations, reducing the clarity of substrate peaks even when gross behavior remains similar. The pattern remains soft but meaningful. When timing holds, lipid oxidation aligns with fasting windows. When timing is perturbed, efficiency can fade. Carbohydrate-linked pathways follow their own daily contours. Proteins that gate pyruvate entry and TCA integration can accumulate at different times than lipid enzymes, biasing mitochondria toward carbohydrate use during specific windows associated with anticipated activity or postprandial states. In skeletal muscle and liver models, circadian control shapes insulin sensitivity, glucose handling, and downstream respiratory support, indicating that carbohydrate metabolism is tuned by both local clocks and systemic timing signals. This layered organization allows glucose-dependent processes to rise when they are most useful, while lipid oxidation steps forward when fasting cues prevail, reducing conflict between pathways and stabilizing redox balance. Underneath these substrate shifts lies a quietly moving redox framework that helps translate time into function. NAD+ cycles, driven in part by clock control of the NAMPT salvage pathway, phase the activity of serine deacetylases that modulate mitochondrial enzymes and antioxidant systems. When NAD+ is higher, mitochondrial SIRT3 and nuclear SIRT1 guide deacetylation programs that can favor fatty acid oxidation and support oxidative phosphorylation with lower byproduct accumulation, especially during fasting-aligned windows. In this way, redox timing complements proteomic rhythms. The proteome provides the broad capacity, and NAD+ sensitive edits provide fine control, together shaping when mitochondria favor lipids or carbohydrates with calm precision. These rhythms are tissue-specific yet coordinated. Liver shows robust daily regulation of lipid and carbohydrate metabolism that responds to both the intrinsic clock and meal timing, while skeletal muscle integrates its own clock with systemic signals to time substrate handling for movement and rest. Cross-tissue studies indicate that clocks in metabolic organs communicate, such that a liver clock can tune rhythmic transcription and mitochondrial respiration features in muscle, aligning inter-organ fuel strategies with feeding cycles. This communication adds stability when liver and muscle clocks cooperate with regular feeding. Fasting, systemic glucose tolerance, and substrate partitioning improve, reducing unnecessary oxidative stress across the network. The role of PER proteins in this orchestration is particularly notable. Quantitative mitochondrial proteomics across the day revealed extensive oscillations in enzymes for lipid and carbohydrate pathways that depend on PER1/2 and substrate-specific respiration peaks that are diminished without them. PER-linked anticipation enables mitochondria to prime for upcoming nutrient states, acting as a rheostat that positions substrate use within healthy redox limits and matched ATP demand. When PER signaling is intact and reinforced by regular meal timing, lipid oxidation crests and carbohydrate processing windows appear in orderly sequence. When PER signaling is absent, these crests flatten and the smooth handoff between fuels is less distinct. Feeding schedule and composition shape the phase and amplitude of these cycles without erasing the principle. Time-restricted feeding can restore or sharpen fatty acid oxidation rhythms under some disruptions, while high-fat diet can dampen temporal control of FAO even when feeding fasting behavior persists, showing that nutrient quality modulates mitochondrial timing programs. In the liver, multiomics data under altered feeding windows demonstrate that phosphorylation, ubiquitination, and acetylation cycles in metabolic proteins remain rhythmic and can be shifted, linking post-translational timing to fuel selection and respiratory readiness. Across these conditions, the imagery of slow-moving switches remains apt. Capacity slides toward one substrate, then glides back, avoiding abrupt transitions that would stress the electron transport chain. Why does this matter for homeostasis at night? During predictable fasting, channeling electrons through pathways poised for lipids and supported by NAD+ aligned defenses can reduce electron leak and manage ROS within signaling ranges, not threat ranges. By separating peak carbohydrate flux from maintenance windows, mitochondria avoid competition between ATP production and repair, permitting mitophagy and biogenesis to proceed with less interference and steadier membrane potential. Over many cycles, the reduced mismatch between supply and demand lowers cumulative oxidative burden and preserves resilience for the next day's activity. A short reinforcement helps the main points settle. Mitochondria favor different fuels at different times, with measured peaks in respiration supported by lipid versus carbohydrate substrates across the 24-hour cycle. These peaks reflect cycling proteomes and PTMs that PCR1/2 and core clock mechanisms help coordinate, aligning fuel choice with feeding, fasting patterns, and expected behavior. NAD+ dependent sirtuin activity provides fine temporal control, shaping enzyme activity, oxidative capacity, and antioxidant readiness to keep electron flow smooth and ROS signaling contained. The overall effect is fewer conflicts between production and repair, calmer redox balance at night, and efficient substrate use by day, achieved through switches that move slowly rather than abruptly. Allow the image to remain tranquil in a dim laboratory of the cell. Quiet hands turn one dial up for lipid oxidation as fasting deepens, and bring another dial forward for carbohydrate handling as activity approaches, all timed by a clock that prefers steady arcs over sharp edges. In the next section, the focus will drift gently to how sleep pressure and mitochondrial state influence each other, and how dynamics and turnover respond when rest is shortened and recovery is needed. Ease into the understanding that sleep need is not only shaped by the brain's electrical networks, but also by the quiet state of its mitochondria, how they handle electrons, how they change shape, and how they coordinate maintenance when wakefulness stretches longer than usual. Recent work in model organisms shows that when sleep is curtailed, mitochondria tend to fragment, mitophagy increases, and contact sites between mitochondria and the endoplasmic reticulum become more numerous, with these alterations softening and often reversing after recovery sleep. The message is gentle but consistent. As wake extends, aerobic metabolism generates molecular signals that raise sleep pressure, and as sleep returns, repair processes follow to restore balance. In carefully controlled studies, sleep deprivation has been observed to shift mitochondrial morphology towards smaller, more fragmented forms in identified sleep control neurons, a change that coincides with increased markers of mitophagy, the selective clearance of damaged mitochondria, and an uptick in mitochondria, ear contact sites that organize both fission and renewal. Using fluorescent reporters, investigators found more mitochondria entering acidic autophagolysosomes after deprivation, indicating active turnover, and more physical opposition between organelles that support lipid transfer, calcium exchange, and the staging of fission events. When animals were then allowed to sleep, mitochondrial shape and network properties rebounded, with volume and branch length returning toward or even above baseline, an orderly rebound rather than an abrupt flip. These structural changes connect to metabolic load. During extended waking, mismatches can develop between electron supply into the respiratory chain and ATP demand, increasing the probability of electron leak and reactive oxygen species formation at the coenzyme Q pool, which in turn promotes fission and primes turnover. In this framework, sleep reduces the mismatch, lowers oxidative strain, and grants time for surveillance and renewal to proceed without competing with peak output. Interventions that alter the flow of electrons, for example, partially uncoupling electron transport from ATP synthesis, can reduce the pressure to sleep, consistent with the idea that aerobic metabolism itself carries a need for periodic recovery when electrons and demand fall out of alignment. A calm but important observation is that mitochondrial dynamics do not merely respond to sleep state, they can also shape it. Experimental biasing of dynamics in defined sleep control neurons shows that promoting fission tends to reduce sleep and blunt rebound after deprivation, while favoring fusion increases baseline and recovery sleep and changes neuronal excitability in directions that match higher or lower sleep pressure. In these same neurons, manipulating DRP1 to drive fragmentation shortened sleep and lowered ATP levels, whereas enhancing OPA1- and mitofuscin-dependent fusion increased sleep and raised arousal thresholds, revealing a reciprocal link between mitochondrial state, cellular energy, and the networks that gate sleep. These effects were cell-type specific, aligning with the notion that sleep circuits host mitochondria tuned to their role in regulating rest and arousal. The increasing contacts between mitochondria and ER with sleep loss has also been seen outside of single neuron studies, with electron microscopy and transcriptomics, indicating that deprivation elevates ER stress signatures and strengthens mitochondria crosstalk in cortex, likely to support higher metabolic throughput and to stage fission and turnover under strain. These contact sites, often termed MAMs, localize the fission machinery and facilitate lipid supply for autophagus and membranes, helping the cell coordinate segmentation, clearance, and eventual rebuilding when conditions allow. Their expansion during sleep loss and normalization after recovery underscores a logistical role, preparing the machinery for repair that will be carried out more fully when wake-driven demand subsides. Together, these lines of evidence support a quiet feedback model. As waking extends, aerobic metabolism increases mitochondrial electron handling demands. If supply and demand drift apart, oxidative byproducts rise, dynamics tilt toward fission, mitophagy increases, and mitochondria contacts proliferate, all of which correlate with increasing sleep pressure. When sleep occurs, membrane potential stabilizes, oxidative load diminishes, and morphology re-equilibrates toward fusion, enabling redistribution of contents and the completion of maintenance tasks that have been staged during wakefulness. This loop is not abrupt. It is a gradual homeostatic adjustment that the cell performs with patience, preserving function while inviting recovery. It is helpful to note that this literature is evolving, and many findings come from model systems designed to isolate mechanisms in well-defined neurons and tissues. Even so, a convergent picture is emerging. Mitochondrial signaling and aerobic respiration contribute to the homeostatic drive for sleep, not as the sole cause, but as an integral component that translates metabolic state into changes in neuronal excitability and network-level sleep need. Reviews and cross-species observations further suggest that sleep promotes mitochondrial redox balance and remodeling, while mitochondrial oxidative status can in turn influence the intensity of sleep homeostasis across age and context. A soft recap may help the ideas settle. Sleep deprivation tends to fragment mitochondria, increase mitophagy, and elevate mitochondria. Contacts. Recovery sleep restores morphology and consolidates repair. Shifting dynamics experimentally toward fission or fusion moves sleep duration and rebound in opposite directions and alters the excitability of sleep control neurons, aligning mitochondrial state with sleep pressure signals. Modulating electron flow can lower or raise sleep need, linking respiratory chain operation directly to the homeostat that governs rest. Across these findings, the tone remains consistent. As wake accumulates, cells prepare for maintenance. As sleep returns, maintenance proceeds and balance is renewed. Let this close gently. It is enough to understand that mitochondria help keep time with wake and sleep, signaling when recovery is due and participating in the repair that follows. Rest allows the quiet work to continue, inspection, clearance, and rebuilding, so that neuronal networks and their energy systems begin the next cycle steady and ready. As darkness settles, a familiar signal rises in the body: melatonin. This hormone is closely tied to night and circadian phase, and its actions extend gently into the landscape of cellular maintenance. In this section, we will move slowly through how melatonin can influence mitochondrial behavior, shaping dynamics, easing oxidative strain, and supporting the processes that clear and rebuild so that the energy system remains calm, balanced, and ready for the next day. The aim is measured and soothing, not to overstate, but to clarify how this nightly signal complements the cell's natural posture of repair. Melatonin is synthesized in the pineal gland with a strong circadian rhythm, increasing in the evening and remaining elevated through much of the night. Many cells also produce melatonin locally, where it can act close to its site of synthesis. Its effects are conveyed through membrane receptors that influence intracellular signaling cascades, and through receptor-independent antioxidant actions that buffer redox stress. Within mitochondria, these influences converge on a few consistent themes: modulation of oxidative stress, support for balanced fission, fusion dynamics, and encouragement of quality control pathways such as mitophagy, particularly under conditions of strain. Begin softly with redox. Mitochondria move electrons through the respiratory chain to produce ATP, and this flow can generate reactive oxygen species as byproducts. Melatonin appears to reduce excessive oxidative stress by several gentle means. It can directly scavenge reactive species. It can upregulate antioxidant enzymes through transcriptional programs, and it can help maintain a redox environment that favors steady electron flow. When oxidative pressure is lower, the mitochondrial membrane potential is more stable, protein and lipid components experience less damage, and the signaling that triggers unnecessary fragmentation is less likely to be engaged. In this way, melatonin provides a calm backdrop against which nightly maintenance can proceed without competing with alarm signals. Now turn to dynamics. The balance between fission and fusion sets the shape of the mitochondrial network and influences how efficiently it distributes membrane potential, how it shares contents, and how effectively it identifies segments for removal. Melatonin has been observed to nudge this balance toward a protective state, limiting excessive fission during stress while preserving the capacity for selective segmentation when needed. This involves modulation of key proteins, DRP1, the GTPs that drives fission, and MFN1, MFN2, and OPA1, which support fusion and inner membrane architecture, through signaling pathways that include protein kinase cascades and sirtuin-linked decetylation states. Melatonin can reduce DRP1 overactivation and support OPA1 integrity, favoring a network that is neither overly fragmented nor rigidly fused. The result is a morphology suited for calm ATP support and readiness for targeted renewal. Mitophagy fits naturally into this picture. When mitochondria are damaged beyond local repair, selective clearance is required to protect the network. Melatonin appears to support mitophagy under stress conditions by stabilizing the signals that mark impaired organelles, such as PINK1 accumulation on depolarized mitochondria, and by facilitating PARK-independent tagging or receptor-mediated routes that recruit autophagosomes. It may also interact with the broader autophagy machinery, helping sustain lysosomal function and the completion of turnover. In practical terms, this means that at night, when maintenance is preferred, melatonin can help ensure that the processes of identifying, sequestering, and recycling damaged components proceed efficiently without tipping the network into unnecessary loss. These effects are not isolated. They are integrated with circadian timing and nutrient state. As NAD+ levels rise in certain nocturnal windows and sirtuin activity increases, melatonin's redox and signaling roles complement acetylation programs that refine respiratory enzyme function and antioxidant capacity. With dynamics balanced, membrane potential steady, and redox defenses prepared, mitophagy can remove what must be cleared, and the cell can then transition smoothly toward biogenesis as dawn approaches. The overall posture is one of coordination. The hormone of darkness aligns with timekeeping signals and metabolic cues to maintain mitochondrial composure through the night. Gently consider the endoplasmic reticulum as well. Contact sites between ER and mitochondria are hubs for fission initiation, calcium exchange, and lipid transfer needed for autophagosome formation. Under strain, these contacts may increase, staging maintenance that is later consolidated during sleep and early recovery. Melatonin's calming influence on oxidative stress and calcium handling can help keep these contact dynamics within a healthy range, sufficient to enable quality control, not so elevated that they reflect unresolved stress. In this way, organelle crosstalk remains orderly, and turnover proceeds without excessive signaling noise. The tone remains reassuring. Too much fragmentation can lower membrane potential and impair ATP delivery. Too much fusion can conceal localized defects and delay clearance. Melatonin's role is not to push the system into a single state, but to preserve balance. Allow fusion to support continuity and efficiency. Allow fission to reveal what needs attention, and allow mitophagy to complete the cycle at a time when it will not clash with high energy demands. When rhythms are aligned, regular light, dark exposure, consistent sleep timing, predictable feeding windows, melatonin's nightly rise fits into a larger pattern that keeps mitochondrial function resilient and calm. A brief reinforcement may help these ideas rest. Melatonin rises at night. It reduces unnecessary oxidative strain, supports a balanced fission-fusion state, and encourages selective mitophagy where appropriate. These actions complement circadian and metabolic signals that set the schedule for maintenance and renewal. In combination, they help preserve membrane potential, smooth electron flow, and keep the network functional and adaptable, ready to meet the next day's needs without carrying excess damage forward. As we transition toward clinical and translational horizons, the themes remain measured. Research continues to explore how melatonin's timing and dose intersect with tissue context, aging, shift work, and metabolic health. Early observations suggest that reinforcing physiological rhythms and protecting mitochondrial balance at night may support long-term cellular resilience. Yet the focus here stays calm and conceptual. Multiple pathways converge each evening: clock genes, NAD plus cycles, sirtuin activity, organelle contacts, and melatonin to maintain the mitochondrial fleet. With night as an ally and signals aligned, the cell's powerhouses move through the dark hours with quiet confidence, repairing gently, conserving steadily, and preparing for light. Settle into a calm understanding that timing is part of cellular care. When circadian cues are steady, light in the day, darkness at night, regular meals, and consistent sleep, mitochondria follow a gentle schedule of fuel handling, dynamics, and renewal. When those cues drift or are inverted, the schedule blurs. In this section, we move softly through what happens when circadian rhythms are disrupted by shift work, irregular light exposure, or genetic alterations in clock genes. The aim is measured and reassuring. Disruption can dampen normal oscillations in mitochondrial morphology, membrane potential, and respiration, yet alignment strategies and gentle routines can help re-establish rhythm over time. The circadian clock speaks to mitochondria through waves of gene expression and post-translational tuning. When clock function is intact, daily patterns appear in fission and fusion proteins, in mitophagy factors, and in respiratory capacity, allowing mitochondria to elongate when continuous ATP support is helpful and to segment when inspection is scheduled. With circadian disturbance, this choreography can flatten. Studies show that core clock gene deficiencies reduce the rhythmic expression of dynamics and mitophagy genes, blunting the normal day-night changes in mitochondrial shape and turnover and contributing to functional decline in tissues such as liver and heart. In practical terms, this means fewer clear windows for maintenance and a greater chance that energy production and repair will compete with one another. A central node in this picture is DRP1, the GTPase that guides fission under healthy conditions. DRP1 activity is tuned across the 24-hour cycle, including rhythmic phosphorylation that helps set when segmentation is favored and when it is restrained. When clock control is impaired, DRP1's rhythms weaken and mitochondrial architecture loses its daily oscillation, often taking on a more persistently fragmented or otherwise abnormal form, with parallel loss of ATP oscillations and reduced bioenergetic flexibility. These changes are not abrupt. They reflect a gradual erosion of timing that normally keeps dynamics and function in balance. Clock genes also shape mitophagy, the selective removal of damaged mitochondria. In aligned conditions, mitophagy regulators, such as PINK1, Parkin, and BNIP3, exhibit time of day patterns that help consolidate turnover during lower demand periods. With clock disruption, expression and cycling of these factors are reduced, and mitophagy intensity falls, leading to the accumulation of dysfunctional mitochondria and diminished respiratory performance, as seen in cardiomyocytes and liver under BMAL1 loss. Quality control still operates, but with less efficiency and fewer coordinated windows, increasing the risk that oxidative stress and low-potential fragments persist. Irregular light exposure and night shift schedules extend these cellular themes into daily life. Human studies associate circadian misalignment with elevated oxidative stress and lower antioxidant defenses, suggesting that when light at night suppresses melatonin and shifts timing cues, redox balance is harder to maintain. While these observations reflect complex behavior and environment, the implication is gentle and clear. When systemic rhythms are disrupted, mitochondria experience more frequent mismatches between electron supply, ATP demand, and antioxidant readiness, which can translate into lower membrane potential stability and increased byproducts. Over time, this can dull the flexibility that usually protects tissues during predictable fasting and activity windows. Not all disruptions are the same. Genetic alterations illustrate how specific clock components anchor mitochondrial timing. Proteins, for example, help schedule substrate preference and mitochondrial respiration. When PER1/2 are disrupted, diurnal peaks in substrate-specific oxidative capacity are reduced and respiratory rhythms flatten. PER mutations can also impair morphology by altering DRP1 regulation, leading to excessive fission, lowered membrane potential, and increased ROS, each of which signals strain on the system. BMAL1 loss, whether whole body or tissue specific, consistently links to diminished respiration, altered dynamics gene expression, and reduced mitophagy, underscoring BMAL1's role in coordinating mitochondrial readiness and recovery. Despite these changes, the message remains reassuring. Mitochondria are adaptable, and circadian systems can re-entrain when light-dark exposure becomes consistent again, when feeding windows are gradually stabilized, and when sleep timing is gently reinforced. Daily patterns in mitochondrial proteins, dynamics, and respiration can regain amplitude. This realignment does not require perfection. It benefits from steady, modest cues, daytime light, evening darkness, regular meals aligned with the biological day, and sufficient sleep that allows maintenance to consolidate. In research models, restoring clock components or reinforcing metabolic timing often improves mitochondrial morphology and function, an encouraging sign that timing is a lever the cell can still use. It may help to anchor these ideas in a calm recap of mechanisms. Dynamics: DRP1 phosphorylation and expression change over the day, and BMAL1/clock linked transcription touches fission and fusion genes. Disruption blunts these cycles, producing abnormal morphology and weakened ATP oscillations. Mitophagy, PINK1, Parkin, and receptor pathways such as BNIP3 normally show rhythmic regulation. Clock misalignment reduces their expression and cycling, decreasing targeted clearance and promoting accumulation of low-function fragments. Respiration and redox: clock deficiencies flatten diurnal respiration patterns and lower oxidative capacity, aligning with higher oxidative stress in misaligned states and reduced ability to match substrate flux with antioxidant readiness. Tissue context: in heart and liver, Clock loss impairs mitochondrial turnover and performance. In immune cells, BMAL1 deletion compromises bioenergetic recovery after inflammatory stress, illustrating that timing supports resilience across systems. The broader health context follows gently from these cellular events. When mitochondrial rhythms are clear, tissues can separate peak output from maintenance, minimizing conflict and preserving function. When rhythms are dulled through shift work, irregular schedules, or genetic alterations, capacity becomes less flexible, redox balance is harder to maintain, and quality control is less synchronized. This does not imply immediate damage, but it suggests a gradual drift toward inefficiency that over long periods may intersect with metabolic and cardiovascular risks observed in misaligned populations. The tone remains steady, these are modifiable patterns, and the system is designed to respond to regular cues. As this section closes, allow the key principle to settle softly. Timing is part of mitochondrial care. When the clock falters, oscillations in morphology, membrane potential, and respiration flatten, fission can drift toward excess, mitophagy can lose precision, and performance can decline in specific tissues. Yet alignment can return with gentle, consistent signals: steady light by day, darkness at night, predictable meals, and restorative sleep, allowing the mitochondrial network to recover its cadence and to separate energy production from repair with the ease it prefers. In the next section, we will move quietly into the immune landscape at night, where mitochondria also keep time, supporting balanced defense and calm readiness across the dark hours. As night unfolds, the immune system does not go silent. It shifts into a calmer mode of readiness, guided by internal clocks that keep time across tissues and cells, including the sentinels that patrol the body's borders and interiors. Immune cells carry their own circadian programs, shaping when they circulate, when they rest, and how their metabolism supports either activation or patient surveillance, with mitochondria helping to set the tone through membrane potential, redox balance, and dynamic structure that adjusts across the 24-hour cycle. In this gentle rhythm, nighttime favors poised maintenance rather than forceful response, allowing energy systems to align with low stimulation periods while keeping defenses alert without strain. Macrophages, which bridge innate sensing and tissue care, illustrate this timing with clarity. Core clock factors, particularly BMAL1, function as metabolic checkpoints that help these cells maintain mitochondrial respiration, redox homeostasis, and controlled inflammatory output when demands change. When BMAL1 programs are intact, mitochondrial metabolism remains steady under stress, preventing excessive reactive oxygen species, limiting HAF1-alpha-driven glycolytic drift, and supporting recovery of oxidative capacity after inflammatory challenges. When BMAL1 is absent, mitochondria fragment more readily, oxygen consumption falls, and succinate-linked OROs rise, amplifying inflammatory signals and blunting the gentle arc of nightly restoration. This timekeeping connects directly to mitochondrial dynamics and potential, which together influence how immune cells balance preparedness with restraint. During low stimulation phases, a more fused mitochondrial network can support stable membrane potential and efficient ATP production for housekeeping tasks, while circadian cues preserve the capacity for selective fission if quality control is needed. In macrophages and related cells, the daily choreography of fusion and fission proteins is supported by clock-linked transcription and NAD+, sensitive post-translational edits, guiding when surveillance proceeds and when renewal should wait for quieter hours. The overall effect is simple and steady, align structure to function so that energy and defense remain synchronized without unnecessary activation. Redox signaling is woven into this nightly posture with care. Reactive oxygen species, when kept within physiological ranges, operate as signals that tune immune pathways rather than as purely damaging byproducts, Circadian regulation of NAD plus metabolism through rhythms in the salvage enzyme NAMT shapes sirtuin activity and downstream desatylation, setting antioxidant defenses and respiratory enzyme function to meet the moment without excess. These NAD plus linked cycles help immune cells maintain membrane potential, manage ROS as inflammation, and avoid oxidative stress during nocturnal vigilance when inflammatory triggers are fewer but not absent. The suprachiasmatic nucleus and peripheral clocks together influence immune traffic, tissue homing, and cytokine balance across the night and day, favoring adaptive initiation and memory consolidation during early sleep, while reserving rapid effector surges for active periods. Within this framework, mitochondria in immune cells follow suit. Membrane potential stabilizes when metabolism matches low external stimulation, and antioxidant systems remain poised, so that a brief demand can be met without destabilizing the network. If a stronger challenge appears, the system is prepared to shift, but the baseline at night remains quiet and efficient, preserving resources for repair and restoration. BML1's role as a metabolic sensor in macrophages shows how timing disciplines immune tone by tempering succinate metabolism and modulating pathways like PKM2, STAT3. BML1 reduces excessive IL-1 drive and preserves mitochondrial function, linking circadian control directly to measured inflammatory output. In its absence, oxidative respiration declines and recovery after stress is delayed, illustrating how the clock supports the transition from activation back to calm readiness, an especially important step during nocturnal maintenance windows. This is not a rigid schedule. It is a gentle guidance that keeps immune metabolism aligned with systemic rest. NAD+ and sirtuin pathways extend this alignment to transcriptional and enzymatic control, shaping both nuclear programs and mitochondrial enzyme states in immune cells as the night progresses. Higher NAD+ availability at specific circadian phases engages SIRT1 and SIRT3, adjusting acetylation of metabolic and antioxidant proteins and refining oxidative capacity while sustaining redox balance. Because these cycles are clock-linked, immune cells experience predictable windows for efficient oxidative metabolism and controlled signaling, supporting mitigation of noise in ROS pathways, while retaining the capacity for decisive response if needed. Endoplasmic reticulum, mitochondria contacts, which can expand under strain to coordinate lipid transfer, calcium signaling, and the staging of fission and autophagosome formation, are also influenced by sleep and circadian state. When wakefulness is prolonged, these contact sites may increase to handle load, but during recovery sleep and aligned nights, they return towards steady baselines that favor unhurried maintenance without continuous stress signaling. In immune contexts, keeping these contacts within a healthy range supports mitochondrial quality control without tipping cells toward activation, sustaining the quiet logistics of turnover when the organism rests. Across the immune landscape, macrophages, dendritic cells, and lymphocytes, circadian immunometabolism provides a shared principle: coordinate substrate use, mitochondrial dynamics, and redox defenses with the expected external environment. Daytime activity is matched to rapid responses and higher flux, while night supports calibration, trafficking to lymphoid sites, and the preparatory steps that underlie efficient future responses. When timing is preserved, mitochondria help maintain this balance by stabilizing potential, modulating ROS as signals, and enabling selective mitophagy and biogenesis to proceed without competing with peak demand. A brief reinforcement may help the ideas settle. Nighttime in the immune system is not inactivity. It is quiet defense, guided by clocks that pace mitochondrial morphology, potential, and redox state towards stability. BML1 and related timekeepers help macrophages and other cells sustain oxidative metabolism under stress and return to baseline with precision, limiting unnecessary inflammatory drive while preserving readiness. NED plus cycles and sirtuin activity translate time into enzyme states that keep ROS signaling informative and membrane potential steady, supporting restorative processes through the dark hours. Hold a gentle image, sentinel cells at ease, their mitochondrial networks elongated enough to keep voltage smooth, their redox systems lit like soft lamps along a corridor, ready to brighten if called, but content to glow steadily in the quiet. In this calm, mitophagy can proceed where needed, and biogenesis can be staged for the morning, ensuring that the machinery of defense is renewed without haste. With timekeeping intact, immunometabolism remains aligned with the night, offering protection that is patient, balanced, and prepared for the day to come. In the quiet hours, communication between organelles becomes especially important, and the conversation between the endoplasmic reticulum and mitochondria is steady and deliberate. Their contact sites, often called mitochondria-associated membranes, serve as logistical hubs where material and signals pass across a very short distance, supporting lipid transfer, calcium exchange, and the organization of machinery needed for fission, mitophagy, and biogenesis. These junctions are not fusions. They are close appositions that allow coordination without losing the separate identities of each organelle. When the system is under strain or when wakefulness extends, these contact sites can become more abundant, preparing the cell to manage load and to stage maintenance that can be consolidated during recovery sleep. These interfaces handle an essential traffic of lipids that the mitochondria cannot synthesize on their own. Phospholipids move from the ER to mitochondria at contact sites, contributing to membrane biogenesis and the renewal of mitochondrial structure, particularly as new organelles are formed or existing ones are rebuilt after turnover. By concentrating enzymes and transfer proteins in a confined microdomain, these hubs maintain efficient exchange and help synchronize membrane composition with the organelle's energetic role. In this way, they are integral to biogenesis, ensuring that fresh membranes are supplied where and when they are needed. Calcium signaling is another quiet duty of these junctions. The ER serves as a regulated reservoir of calcium, and brief, localized transfers to mitochondria at contact sites can stimulate oxidative metabolism and support ATP production. When tuned appropriately, these calcium handoffs enhance bioenergetic readiness without overwhelming the organelle, allowing mitochondrial enzymes to respond to small, timely pulses. Under early adaptive stress, increased contact can facilitate calcium transfer that boosts mitochondrial metabolism, helping cells meet demand while maintaining orderly control of redox status and unfolded protein responses. Fission is often staged at these ER mitochondria interfaces. The ER can wrap partially around a mitochondrial tubule and mark a constriction point, where the fission machinery assembles to complete division. DRP1, the principal mechanoenzyme for mitochondrial fission, is recruited to these marked sites, aided by adapter proteins, membrane lipids, and local curvature that favor assembly. This choreography ensures that segmentation is placed precisely, allowing selective isolation of regions that need inspection or removal. The ER's positioning helps couple membrane remodeling to downstream quality control pathways that can proceed more efficiently once the fragment is separated. These contact sites adapt with behavioral state. Studies show that acute sleep loss increases ER mitochondria contacts in cortical neurons, alongside transcriptional signatures of ER stress and mitochondrial activity. This expansion likely supports higher metabolic throughput during extended wake and positions the fission and turnover machinery for later use. Electron microscopy reveals more frequent contacts after deprivation, while gene and protein markers of contact and calcium handling rise in parallel. With recovery, sleep, morphology, and contact patterns shift back toward baseline, suggesting that these structural adjustments are part of a reversible logistics plan that prepares for repair when the organism can rest. In identified sleep control neurons, extended wakefulness similarly elevates contact counts and mitophagy markers, with fission features appearing first and a rebound in mitochondrial number, volume, and branching during recovery sleep. This sequence, contact site increase, fission and turnover, then fusion-supported rebuilding, illustrates a calm temporal choreography. Stage segmentation at ER-marked sites, remove what is beyond repair, then restore network continuity and capacity as the night yields to recovery windows. Contact sites thus act as both staging grounds and supply depots, coordinating lipids for new membrane, positioning fission machinery, and facilitating the autophagic handoff. The lipid environment at these interfaces also tunes the mechanics of fission. Mitochondrial outer membrane lipids, including cardiolipin and diacylglycerol, can modulate DRP1 recruitment and activity, while ER shaping and curvature help define constriction zones. Biophysical studies show that DRP1 interacts with cardiolipin in ways that enhance its GTP's function under specific membrane conditions, and that local lipid defects or cone-shaped species can promote efficient constriction. Such refinements suggest that the mitochondria platform not only brings proteins together, but also crafts a membrane landscape favorable for precise division, minimizing unnecessary fragmentation and preserving function. Through the night, these contact sites help separate high-output tasks from maintenance. When energy demand recedes, fission events at ER-marked positions can isolate low-potential regions for mitophagy, while the ER supplies lipids and helps assemble autophagosomal membranes to complete turnover. Later, as biogenesis ramps under transcriptional programs, mitochondria lipid exchange supports the incorporation of fresh membranes and the assembly of new respiratory units, with fusion restoring network continuity. All of this occurs in measured steps, paced by circadian cues and supported by redox-linked timing signals, so that production and repair do not compete. When cues are disrupted, contact dynamics can lose their gentle rhythm. Sleep deprivation increases contact density and ER stress signatures, indicating a compensatory response to load that, if prolonged, may strain redox control and calcium handling. Yet the system remains plastic. Recovery sleep and re-established timing reduce contact abundance toward baseline and allow morphology to recover, reflecting the reversible nature of these logistics. In experimental models, promoting contacts can alter sleep architecture, further linking these hubs to the homeostatic adjustments that follow wake. A brief reinforcement may help the ideas settle. ERs and mitochondria contact sites are logistical hubs for lipids and calcium, and assembly points for fission and subsequent quality control. Under strain or extended wake, their number increases, staging segmentation and turnover. With recovery, sleep, fusion and biogenesis complete the cycle, supported again by lipid exchange at the same hubs. This choreography is paced by temporal programs that keep maintenance aligned with low-demand periods, preserving membrane potential and metabolic stability across cycles. Hold a quiet image as this section closes, two organelles meeting at narrow bridges, passing small parcels of lipids, brief pulses of calcium, and placing careful markers where division should occur. The handoffs are slow and deliberate, neither hurried nor withheld. Through the night, these bridges help the cell keep order, fission before clearance, then fusion and rebuilding, so that by morning the energy system is steady, renewed, and ready for the day's work. Settle into a calm overview of how the mechanisms we have explored express themselves in health and disease, especially in the heart and across the arc of aging. The focus is steady and measured. PGC1-alpha as a central coordinator of mitochondrial capacity and renewal. The importance of balanced fission and fusion for bioenergetics and quality control, and how moderate, well-timed activation of biogenesis and mitophagy supports long-term resilience. The aim is to connect principles, not to prescribe, highlighting how rhythmic maintenance, guided by upstream sensors, preserves function in tissues that depend deeply on oxidative metabolism. Begin gently with PGC1-alpha. This co-activator sits at a key metabolic node, integrating signals from energy sensors to drive mitochondrial biogenesis and functional capacity through partners such as NRF1, NRF2, ER-alpha, and PPAR pathways. coordinating nuclear and mitochondrial gene programs for respiratory complexes and mtDNA care. When activated by AMPK-mediated phosphorylation and SIRT1-mediated desethylation, PGC, 1-alpha promotes transcription of oxidative phosphorylation subunits, TFAM, for mtDNA replication and transcription, and protein import machinery, raising oxidative capacity in a phased and organized manner. This axis ties biogenesis to quality control, ensuring that increases in capacity are matched by the maintenance of genome integrity and proper assembly of respiratory units. The heart illustrates this coordination with particular clarity. Cardiomyocytes rely heavily on fatty acid oxidation and robust mitochondrial respiration. PGC, 1-alpha, is highly expressed in myocardium and supports the postnatal shift toward oxidative metabolism and the sustained expression of FAO and OXPHOS genes through PPAR-alpha and NRF-dependent programs. When cardiac demand is high, or when development requires a new energetic set point, PGC, 1-alpha helps expand and refine mitochondrial function, maintaining fuel flexibility and ATP delivery with stable membrane potential. In models of cardiac stress, reductions in PGC, 1-alpha associate with diminished biogenesis, altered dynamics, impaired mitophagy, and energy deficits. Conversely, restoring PGC, 1-alpha can enhance mitochondrial morphology, respiration, and mitophagy flux, supporting cardiac homeostasis under physiological conditions. Quality control depends on dynamics, mitochondrial fission and fusion shape network continuity, segregate damage, and influence calcium handling and redox balance. Imbalances in either direction are linked with cardiac dysfunction and aging phenotypes. Excessive or persistent fission is associated with fragmentation, lower membrane potential, and increased oxidative stress. Excessive fusion can mask damaged regions and impede selective mitophagy, allowing dysfunctional components to persist. In the heart, disruptions to mitophysins, MFN1/2 or OPA1, produce characteristic deficits in mitochondrial structure and autophagic flux. With age, while unchecked fission drivers exacerbate dysfunction and stress signaling, a more balanced restoration of both fission and fusion yields better outcomes than targeting only one arm. These dynamics interface with mitophagy and biogenesis. In healthy tissue, fission helps isolate low-function segments for removal, while PINK1, Parkin, or receptor pathways, for example, BNIP3/FUNCS1, coordinate clearance. Biogenesis then replenishes capacity, guided by the PGC, 1-alpha. PGC, 1-alpha also links to mitophagy control through direct transcriptional influence on components like PINK1 or FUNCS1, and through ERR-alpha SIRT3 interactions, helping align removal with replacement, so the pool of mitochondria remains functional over time. This coupling supports stress resistance. When clearance rises, for example, after strain, renewal follows, preventing attrition of ATP output and limiting the accumulation of oxidized proteins or mtDNA damage. Across aging, these themes converge. The aging heart shows accumulation of dysfunctional mitochondria, altered permeability transition behavior, calcium mishandling, and changes in dynamics proteins, often with giant damaged mitochondria and cristae disruption, alongside shifts in MFN, OPA1 and DRP1 expression patterns. Mitophagy commonly declines, which allows damaged organelles to persist, selectively restoring turnover, and rebalancing fission and fusion can reduce senescent features, improve bioenergetics and support function in models. Genetic and experimental evidence suggests that both insufficient fusion and unchecked fission can impair cardiac performance, whereas coordinated normalization, rather than one-sided modulation, helps preserve contractile function and mitochondrial quality as organisms age. PGC, 1-alpha, remains central in this context. As a nodal regulator of mitochondrial biogenesis and oxidative capacity, It integrates AMPK and SIRT1/NAD plus signals to maintain a resilient energetic profile and to prime antioxidant defenses during periods of increased respiratory activity. In cardiovascular disease models, reduced PGC-1-alpha correlates with energy failure and maladaptive remodeling, while appropriately scaled increases in PGC-1-alpha activity can normalize respiration and morphology and support mitophagy, buffering against progression when overall cardiac structure is intact. Because sirtuin pathways modulate both PGC-1-alpha and mitophagy regulators, NAD plus availability emerges as a quiet lever that links feeding, fasting state, circadian timing, and mitochondrial quality control during aging. Clinical horizons are measured and still developing. Preclinical work suggests that moderating excessive fission, supporting fusion integrity, and restoring mitophagy can improve mitochondrial quality and cardiac outcomes, while interventions that broadly enhance PGC-1-alpha mediated biogenesis need to be carefully titrated to avoid imbalances in organelle proliferation versus function. Early strategies aim to reduce fragmentation, stabilize OPA1 processing or fine-tune DRP1 activity, and to reinforce mitophagy where it has waned, with the goal of improving ATP supply, lowering RO's burden, and protecting calcium homeostasis in ischemic and failing hearts. Translational challenges remain: tissue specificity, dosing, timing with respect to circadian phases, and long-term safety all require cautious evaluation before broad clinical application. The broader metabolic picture reinforces the value of rhythmic maintenance. Systems that keep NAD plus cycles, sirtuin activity, and PGC-1-alpha programs aligned with predictable day-night patterns reduce mismatches between demand and capacity, lowering oxidative stress and preserving membrane potential stability over many cycles. Rhythmic alignment, regular light, dark exposure, consistent feeding windows, and preserved sleep supports mitochondrial proteostasis and turnover, offering a non-alarmist path to maintain function alongside pharmacologic avenues that may target dynamics or biogenesis directly. Aging tissues appear to benefit from this layered approach. Gentle restoration of mitophagy, balanced dynamics, and PGC-1-alpha guided renewal can together sustain resilience longer than any single lever in isolation. A brief calm synthesis may help the ideas rest. PGC-1-alpha occupies a central node that connects AMPK and SIRT1/NAD plus signals to mitochondrial biogenesis, respiratory capacity, and coordination with mitophagy via NRF-driven programs and TFAM-supported mtDNA care. In the heart, where energy demand is high, this axis helps maintain FAO and OX-phos, stabilizes morphology, and supports turnover when it falters. Bioenergetic flexibility declines and remodeling risks increase. Balanced fission and fusion protect membrane potential, segregate damage, and enable selective clearance. Imbalances in either direction are linked with cardiac dysfunction and features of aging, while coordinated normalization improves outcomes in models. Moderate, well-timed activation of biogenesis and mitophagy, supported by circadian and metabolic cues, appears to contribute to healthy aging by reducing cumulative oxidative burden and preserving ATP delivery under variable demand. Let the takeaway be simple and reassuring. Mitochondrial health in clinical contexts reflects the same principles seen at night in the resting cell: rhythmic maintenance, balanced dynamics, and coordinated renewal. PGC-1-alpha and its upstream regulators provide the transcriptional and enzymatic timing, while fission, fusion, and mitophagy execute the logistics of care. Together, they sustain cardiac homeostasis, support metabolic health, and buffer the effects of aging, not through forceful intervention, but through steady alignment of capacity with need, cycle after cycle. Settle into a calm picture of the day, arranged by simple cues: light and dark, feeding and fasting, activity and rest. Within this predictable pattern, mitochondria listen to nutrient state and time, adjusting capacity with quiet precision. When meals occur at regular times and nights are left undisturbed by food, energy sensors and transcriptional partners find their cadence, and renewal processes are scheduled without competing with peak demand. This section offers a soft map of those connections, how feeding-fasting cycles speak through AMPK, NAD plus linked sirtuins, and PGC-1-alpha to guide respiratory genes and the timing of biogenesis, without prescribing behaviors and always keeping the tone unhurried. Begin with the idea of fasting as a gentle signal. Hours without nutrient inflow shift the cellular energy ratio toward a state sensed by AMP, a kinase that notices when ATP is being used, and AMP rises. When AMPK is engaged, it softly pivots the cell toward efficiency, promoting catabolic pathways that generate ATP, easing back on anabolic tasks and opening doors to maintenance. In mitochondria, this includes enhanced fatty acid oxidation, careful handling of electrons, and an increased readiness for quality control. As evening lengthens, a defined fasting period provides time for these processes to find rhythm, separate from the daytime focus on immediate energy supply. Feeding, in turn, carries its own timing information. When meals arrive in consistent windows, cells anticipate substrate availability, and the circadian program aligns transcription to match. The same pathways that respond to nutrients also touch the clock's machinery, so that metabolic cues do not compete with timekeeping, but rather reinforce it. In this way, substrate choice and respiratory throughput can crest at appropriate hours, while maintenance is gently diverted to quieter phases. The body prefers these patterns. They reduce mismatches and keep redox balance steady. PGC-1-alpha sits at the center of the renewal plan. It is a co-activator that partners with transcription factors to increase the expression of mitochondrial respiratory genes, import machinery, and factors that care for mitochondrial DNA. Its activation is shaped by the energy landscape. AMPK can phosphorylate and enhance PGC-1-alpha's activity when energy is low, while the NAD plus dependent decetylase SIRT-1 refines PGC-1-alpha's function when NAD plus is abundant. Because NAD plus availability and AMPK tone follow feeding, fasting, and circadian programs, PGC-1-alpha receives a quiet schedule, windows when biogenesis is favored, often following periods of selective clearance, so that new capacity is installed without burdening the network during high demand. NAD plus is a subtle timekeeper in this conversation. Its levels rise and fall across the day, reflecting both the clock's control of its salvage pathway and the cell's metabolic state. When NAD plus is higher, sirtuins such as SIRT-1 in the nucleus and SIRT-3 in mitochondria become more active, decetylating proteins that influence respiratory flux, antioxidant defenses, and transcriptional control. This provides a second layer of timing on top of proteome changes, fast, reversible edits that align enzyme behavior with phase. In fasting-aligned windows, these edits tend to support oxidative efficiency and protect membrane potential, allowing maintenance to proceed in the background. Meal timing acts as a soft trainer of peripheral clocks. When feeding occurs at consistent times, local clocks in metabolic tissues align their gene expression cycles to match, without disturbing the central pacemaker that follows light. This entrainment places metabolic peaks and troughs in order carbohydrate handling when activity is expected, lipid oxidation during fasting, and quality control when demand is lower. If meals shift to unusual hours, peripheral rhythms can drift, and the fine structure of mitochondrial oscillations may blur. Returning to regular windows helps the system find its phase again slowly and reliably. Within this map, AMPK serves as a courteous switchboard. In fasting or energetic strain, it signals that efficiency is needed, nudging cells toward fatty acid oxidation and enhancing autophagic readiness, while also promoting transcriptional programs that favor renewal through PGC. At the same time, AMPK's influence reaches into the clock, helping sustain the oscillations in molecules that carry time into enzyme states. The effect is not forceful. It is a series of small adjustments that together create a dependable rhythm. Respiratory gene expression follows these cues in waves, not steps. When PGC1-alpha is engaged in the appropriate phase, transcripts for oxidative phosphorylation and mitochondrial maintenance rise in an organized fashion, and factors like TFAM prepare mitochondrial DNA for accurate replication and expression. Later, when feeding resumes and energy is plentiful, these programs taper and mitochondria shift back to supporting immediate demands, with maintenance temporarily in the background. The benefit is separation of tasks, production in one window, repair in another, renewal where it fits best. This separation also steadies redox balance. When feeding and fasting occur predictably, substrate flow through the respiratory chain is smoother, membranes maintain potential with less fluctuation, and antioxidant systems keep pace without abrupt surges. Reactive oxygen species remain within signaling ranges, useful for guiding adjustments rather than triggering distress. Over many cycles, the cumulative burden lightens and the network's flexibility is preserved. It is helpful to remember that tissues vary. Liver, muscle, and heart each carry their own clocks and respond to feeding, fasting with distinct amplitude and phase, yet they share the same logic. Align intake with expected activity, maintain a nightly period for inspection, and schedule biogenesis toward the transition back to day. When system-wide cues are coherent, light, meals, rest, these tissues coordinate and mitochondria across the body find the same regularity. Now, allow a brief reinforcement. Predictable fasting and feeding act as quiet teachers of time, shaping AMPK tone, NAD+ cycles, and sirtuin activity. These in turn activate and refine PGC1-alpha, lifting respiratory gene expression and mitochondrial renewal into appropriate windows. The result is a calm choreography, substrate choice aligned with demand, quality control with fasting, and biogenesis as night gives way to morning. Nothing abrupt, nothing forced, just the body's preference for patterns and the mitochondria's responsiveness to those soft cues. As this section closes, keep the conceptual map simple. Regular meals and restful nights keep clocks aligned. AMPK notices energy state and opens space for maintenance. NAD+ carries time into enzymes, and sirtuins translate it into gentle edits. PGC1-alpha coordinates the rebuilding that follows clearance. Together, they guide mitochondrial capacity to where it is needed, when it is needed, across the cycle of day and night. In the next section, we will weave these pieces into the full life cycle, how damage sensing, selective clearance, and biogenesis connect across time to preserve the network with steady care. Let this section bring the pieces together into a single quiet arc. Over each day and night, mitochondria move through a life cycle guided by time. They sense and segregate damage, remove what cannot be repaired, and then restore capacity through carefully timed renewal. The sequence is not abrupt. It follows circadian cues that place each step into windows that minimize conflict with activity and align with the ebb and flow of nutrients and energy demand. We will revisit the key players: DRP1, MFN1, and MFN2, OPA1, PINK1, PARKIN, PGC1-alpha, NRFs, and TFAM, and follow their relay across the clock, keeping the focus on coordination rather than isolated events. Begin with sensing and segregation. Mitochondrial dynamics provide the first layer of triage through fission. Guided by the GTPase DRP1, the network can separate regions that show lower membrane potential, disrupted respiration, or oxidative damage. This is not indiscriminate cutting. It is staged at sites often prepared by the endoplasmic reticulum, where contact points mark constrictions and help assemble the fission machinery. In parallel, fusion, supported by MFN1 and MFN2 on the outer membrane and OPA1 on the inner membrane, maintains continuity, sharing contents, and stabilizing cristae structure. When the network is more fused, membrane potential is easier to distribute and small defects can be buffered. When surveillance is needed, a modest tilt toward fission allows the cell to isolate segments for inspection. The circadian system helps set this balance through rhythmic changes in expression and post-translational states of dynamics, proteins, ensuring that segregation is most active when it will not compete with peak ATP demand. Once segments are isolated, the cell decides what can be preserved and what must be cleared. If a fragment's membrane potential remains low and local repair does not restore function, mitophagy pathways engage. One well-characterized route begins with PINK1, which accumulates on the outer membrane of depolarized mitochondria. There it phosphorylates ubiquitin and parkin, an E3 ligase, recruiting parkin to deposit ubiquitin tags on outer membrane proteins. These tags are read by autophagy receptors that connect the marked mitochondrion to the forming autophagosome, which then delivers its cargo to lysosomes for degradation. Receptor-mediated mitophagy can also proceed via BNIP3, NICS or FUNCS1, particularly under hypoxia or stress, with phosphorylation codes that align removal with cellular conditions. The choice of pathway is context-dependent, but the purpose is consistent: remove what cannot be safely returned to the network and recycle components. Circadian timing supports this clearance by arranging complementary conditions during low-demand windows, often in the night for diurnal organisms. NAD+ availability rises and sirtuin activity increases, refining antioxidant defenses and tuning respiratory enzymes to a calm state. Dynamics lean gently toward inspection readiness, and autophagy gene expression is more favorable for turnover. In these conditions, mitophagy can proceed without drawing heavily on ATP reserves needed for activity. When sleep follows extended wakefulness, mitochondria contacts that staged fission during strain help complete the logistics of clearance, and markers of turnover increase, consolidating repair. After removal comes rebuilding. PGC-1-alpha, the central cull activator of mitochondrial biogenesis, is brought forward by signals that integrate energy state and circadian phase. AMPK, sensing higher AMP-SAE-TP, and SIRT1, sensing higher NAD+, activate PGC-1-alpha through phosphorylation and decetylation. PGC-1-alpha then partners with NRF1 and NRF2 to raise the expression of nuclear genes encoding respiratory chain subunits. Import machinery and the mitochondrial transcription factor TFAME. TFAME supports mitochondrial DNA replication and transcription, ensuring that new organelles carry intact genetic instructions and that the dual genomes, nuclear and mitochondrial, remain synchronized. This transcriptional wave is timed to follow clearance, so capacity is restored steadily, not in a way that conflicts with immediate energetic needs. There is cross-talk between removal and renewal that keeps the cycle coherent. Parkin-dependent mitophagy can relieve repression on PGC-1-alpha by targeting its inhibitors for degradation, gently coupling the act of clearing damaged mitochondria to the permission to rebuild. Conversely, PGC-1-alpha and its transcriptional partners can influence the expression of mitophagy-related genes, ensuring that the system can respond if quality control needs remain. The effect is a relay: dynamics segregate, mitophagy clears, transcription restores, and fusion helps re-establish network continuity, with each step primed by the last and guided by the clock. Across this sequence, redox and membrane potential carry quiet signals that reinforce timing. Oscillations in NAD+ translate feeding, fasting patterns into sirtuin activity, which adjusts acetylation states of respiratory and antioxidant proteins, preserving efficiency during maintenance windows. When substrate flow is aligned with capacity, electron transport proceeds with fewer detours, membrane potential stays within a stable range, and reactive oxygen species remain in informative rather than damaging concentrations. This environment makes both clearance and rebuilding safer, less collateral oxidation during turnover, and smoother assembly of respiratory complexes during biogenesis. The mitochondria interface serves as a logistical hub throughout. Contact sites increase under strain, helping position fission events and supplying lipids for autophagous formation. As repair proceeds and biogenesis begins, the same interfaces support membrane renewal for new or expanded mitochondria. Calcium exchanges at these sites are kept within physiological pulses that support metabolism without overwhelming the organelle. With recovery sleep, contact density and stress signatures subside toward baseline, reflecting the completion of the night's logistics and the return to steady readiness. It may be calming to walk through the clock's roll once more, briefly and in order. Clock and BMAL1 initiate daily gene expression waves that include metabolic and quality control factors. Cursor and cry feedback to set phase and amplitude. These clock components touch dynamics, mitophagy and biogenesis through both transcriptional programs and NAD+ linked post-translational control. In response, mitochondria adopt a morphology suited to the moment, clear what is damaged when energy demand is lower, and rebuild as dawn approaches, with PGC1-alpha, NRF1/2, and TFAM guiding the renewal. The result is a daily cadence in which energy production and repair are separated just enough to reduce friction. This organization carries practical consequences for resilience. When the cycle is intact, regular light, dark cues, consistent meal timing, and sufficient sleep, mitochondria avoid prolonged mismatches between supply and demand. Damaged fragments do not linger, turnover is neither rushed nor delayed, and renewed capacity meets the next day's requirements with minimal oxidative burden. If timing is disrupted, the relay can blur, fission may outpace mitophagy, or biogenesis may arrive out of phase with clearance, leading to persistent fragmentation, lower membrane potential, and reduced flexibility. Reestablishing cues helps restore the sequence, often gradually but reliably. A soft recap settles the picture. DRP1 guided fission and MFN/OPA1 supported fusion detect and manage variability in mitochondrial health. PINK1, Parkin and receptor pathways carry out selective removal when local repair is not enough. PGC1-alpha, activated by AMPK and SIRT1 and partnering with NRFs and TFAM, replenishes capacity by coordinating nuclear and mitochondrial genomes. NAD+ rhythms and sirtuin activity refine each step, while mitochondria context provide the logistics of division, turnover, and membrane supply. All of this is paced by the circadian clock, which schedules maintenance into the quieter hours so that production and repair rarely compete. Hold a gentle image as we close. A fleet of cellular power plants moving through the night in measured steps, segments set aside for service, worn parts replaced, lines checked, and capacity restored before morning. The night is not merely rest, it is scheduled care. With the clock as a guide, the life cycle repeats calmly, preserving function for the day to come. In the final section, we will let these themes drift into a quiet recap of the night's work and how this steady maintenance supports tomorrow's energy. Let this closing section settle in slowly, like the last few breaths before sleep. Across the night, mitochondria follow a measured rhythm. They tune respiration to the hour, keep redox signals within comfortable ranges, reshape their networks through fission and fusion, and coordinate careful renewal. The circadian clock guides this choreography, placing each task into a window where it fits best: activity when it is needed, inspection when it is calm, rebuilding when the new day approaches. Nothing is rushed, nothing is forced. It is a cycle that repeats with quiet reliability. Begin with respiration. Oxygen use and electron flow through the respiratory chain rise and fall over the day, with substrate preference shifting in time, carbohydrates favored in some windows, lipids in others. These changes are not abrupt switches, they are slow waves, aligned by clock signals and nutrient timing that keep ATP supply matched to demand. When the rhythm holds, the electron transport chain works efficiently, membrane potential remains steady, and byproducts are easier to manage. Over many nights, this gentle matching lowers the cumulative oxidative load. Redox balance moves with the same calm tide. NAD+ availability follows a daily cycle influenced by the clock and by feeding-fasting patterns. As NAD+ rises, sirtuins adjust protein states, within mitochondria through SIRT3 and within the nucleus through SIRT1, fine-tuning respiratory enzymes, antioxidant defenses, and transcriptional programs. These small edits help maintain stability, smooth electron flow, stable potential, and reactive oxygen species kept within informative, non-disruptive ranges. Through the dark hours, this chemistry becomes a quiet lantern, lighting the way for maintenance without waking the system. Network shape supports these goals. Fission and fusion alternate like gentle steps, guided by DRP1, MFN1/2, and OPA1. When continuity is useful, fusion spreads membrane potential and shares components across the network. When inspection is due, fission isolates segments that need attention. The circadian system nudges this balance with subtle changes in protein expression and modification, letting surveillance proceed when demand is low and continuity return as morning nears. The effect is a steady morphology that protects function and prepares for renewal. Quality control completes the picture. When segments are too worn to repair, mitophagy quietly removes them. PINK1 and PARKIN mark impaired mitochondria. Receptor pathways stand ready under specific conditions. Autophagosomes enclose what must be recycled. Lysosomes complete the task. Endoplasmic reticulum, mitochondria contact sites serve as patient logistics hubs, placing fission, exchanging lipids, and coordinating calcium at the right moments. After clearance, transcriptional programs rise under PGC, 1-alpha, partnering with NRFs and TFAM to restore capacity, maintain mitochondrial DNA, and assemble new respiratory units with careful timing. Renewal often meets dawn, so the day begins with a refreshed, stable pool. These elements form a feedback loop with sleep. Extended wakefulness raises metabolic pressure and can fragment networks, increase contact sites, and prime turnover. Sleep allows the opposite stabilization of membrane potential, calm redox, and completion of maintenance that was staged during the day. Recovery sleep brings morphology back toward fusion, consolidates mitophagy, and makes room for biogenesis. As this cycle repeats, performance resets and oxidative burdens are kept small. The loop is gentle. Metabolism shapes sleep need. Sleep supports mitochondrial care. Mitochondrial signals help the system know when rest is due. The clock is the thread that keeps the pattern intact. Clock and BMAL1 initiate daily waves of gene expression. PERD can cry, refine the phase. NAD+ carries time into enzymes. Sirtuins translate it into protein states. Downstream, mitochondria read these cues. Respiration adjusts. Dynamics shift, quality control proceeds, and biogenesis follows. When rhythms align, regular light by day, darkness at night, consistent meals, and time for sleep, the cycle gains clarity. When rhythms falter, the peaks and troughs flatten, and the separation between production and repair blurs, yet the system remains adaptable. With gentle cues, cadence returns. It may be helpful to let the main themes drift by once more, unhurried. Mitochondria are rhythmic power plants. They tune oxidative capacity to time of day. They balance redox through NAD+ cycles and sirtuin activity. They reshape their networks to distribute energy or to prepare for selective clearance. They remove what cannot be fixed and rebuild what is needed, guided by PGC, 1-alpha, NRFs, and TFM. Mitochondria context organize the logistics. Sleep provides the quiet interval in which maintenance can complete. With the clock in place, these steps occur in sequence, preventing conflict between immediate performance and long-term care. If attention has wandered, that is welcome. This content is meant to be absorbed softly. The exact terms, oxidative phosphorylation, membrane potential, mitophagy, biogenesis, have appeared and reappeared, each time with a little more detail, and always with the same message. Timing matters, balance matters, and the cell prefers calm routines. Even under stress, the default is orderly cleanup and measured renewal. Let the closing be simple. The night's work is careful and consistent. Respiration is smoothed. Redox is steadied. Networks reshape and are restored. Damaged parts are removed. New capacity is readied for morning. Sleep, metabolism, and mitochondrial signals cooperate to reset performance and reduce strain. As the next day approaches, the system is prepared, not by force but by rhythm. It is enough to rest. The cells know their work.